All right. So our next session of talks are our innovation session. So we um, reached out to all our vendors to get some feedback from them and hear about their new technologies that they have to share. So our next few talks will be on some technologies that you might not be aware of. So we're just highlighting those. And our first speaker is Alex Hyler from Cyto Recovery. Her talk is titled Label-Free Cell Enrichments on Cyto R1, Improving the Viability, Sustaining Phenotype, and Maximizing Cell Recovery. Dr. Hyler earned her PhD in biomedical engineering from Virginia Tech Wake Forest Biomedical Engineering and Sciences in 2018. Wow, that's a mouthful. Sorry. Which is, yeah, wow. Wow. And she also did a Fulbright conducting research in Denmark. She was trained in some really cool laboratories in Virginia Tech and has expertise on ovarian cancer biology, microfluidics, and device engineering. As Cyto Recovery's chief science officer, Alex has been with the company since its founding in 2018 and currently manages the company's operations, including product development, R&D, manufacturing, and sales. Alex earned her bachelor's with honors in chemical engineering from the University of Kansas in 2013. But when she's not busy working, which is probably all the time, she does like to travel, watch sports, and play with her dogs and spoil them rotten. Not surprising. Take it away.
Well, thank you. Thanks, guys. I know I'm the first talk post-lunch. Good news, bad news, I talk really fast, so I'm going to wake you right back up. But many of you have come by our booth today and said, Cyto Recovery, never heard of it. You don't offend me. We are brand new into the space, and I'm going to challenge you today. You guys already know how exciting and important. Let's make sure we're on the slide deck. There we go. You already know how important cell sorting is, so I love conferences like this. I don't have to justify why we cell sort. I always open my talks with, you don't wake up and think about cell sorting like I do every day, but fun fact, most of you actually do. And you know how important it is to do precision cell recovery, specifically in areas like discovery in the cancer continuum, stem cell therapy development, and single cell genomics and analysis, 3 of our target markets. But not all sorters do all the things, right? There's a reason you guys have flow cores, you have 8 instruments, 7 instruments, 5 instruments, right? And some of the challenges today involve high sample losses and high cell death, right? So those jet nozzles, not all cells love that. As you label the cells, you can often change their behavior. And of course, you need antibodies, right? You're going to have to put them through and have some sort of tag to actually sort based on. So this is our benchtop instrument that starts solving some of those problems and provides you another tool in your toolbox. We can do really small cell sample inputs. I'll show you data on that today. We have high viability recovery, and we preserve the phenotype, meaning the cells go in and come back out the exact same way. And then the best part, it's all done label free. So we're not going to antibody tag anything. I'm going to do it all based on just how the cells are in their native state. So our product lines include the actual benchtop instrument, and I'll talk about the options for that. It looks like this. It takes about 3 feet of bench space. Can also be put in a hood if you're worried about that. Big black box on the bottom left provides electronics. I'm going to explain to you why you need electronics. On top is how you introduce your sample onto our little micro devices, which I'll show you in a second. The microscope could be any microscope. I use a Bico one here. I don't care what scope you put it on. It's agnostic. But what we do want to do is visualize what's happening, right? Cursive label free. If I don't have a fluorescent marker, I don't know how effective your sort is. So I'd like to visualize and make sure it's working. And then it's all driven by software. And then on top of that, there are disposables, just like any other tool. So we have our disposable kits. Everything your cells touch that you see on the left all the way through to collection on the right is a one-time disposable kit, meaning it gets thrown away. You can run your cancer microbiome in the morning, your patient samples in the afternoon, and never have to worry about bleaching or dealing with any sterility concerns because everything the cells touch gets put away and thrown away. We have a reagent line customized to different cell types and applications, and as I mentioned, the software-driven database. Okay, let's talk about the really cool part. How do I actually do this, right? You just told me you label free sort, explain it to me. Micro device looks like this, little tiny thing in my hand. Inside of it, what you can't see are these 20 micron pillars, okay? So imagine the Plinko board game, if anyone ever played that game back in the day. And as the cells go through, we turn on those electrodes, those red things on the side, and the cells act like little hot spots. So they are not magnetic, but they act like a magnet now. And certain cells attract towards those pillars while the rest of your population continues to sort through. So here in this example, we're trapping the red cells. Maybe those are the ones I care about. Maybe they're in the way, right? It can work either way. You can do negative or positive selection, but it's all fully reversible, and you can collect off your subpopulations after you have enriched using their electrical signature. Okay, cool. You said electrical signature multiple times. What does that mean? Great question. I wish I could tell you it was really simple, like it was just based on size, or it was just based on their membrane capacitance or something like that. It's unfortunately a very complex problem inherent to each cell type. But as you can see here, by tuning frequency, which is here on the y-axis, and voltage, instead of tuning your lasers, you can get different cells to have different responses, right? We were invented down here at the bottom with ovarian cancer and ovarian cancer stem cells back when no one knew what a cancer stem cell was, didn't have any labels for it. Turns out biologists said, these ones are weirder, help me find them, right? So the engineers came in and helped them find them. Now we do everything head to toe, literally neurons all the way down to sperm on the platform, and I'm going to show you some of that data. But importantly, because we do a lot of different applications, we can customize the geometry. So these little micro devices come in a whole bunch of different geometries to accommodate your needs. Our A, B, and C chips are variations of electrode, pillar size, spacing, right? We don't want any clogging. We can do any cell size. We can do all the way down. We've done all the way down to bacteria, and we're starting to tackle other EVs and smaller particles too. So we can adjust that for batch-based collection. And then we just introduced the continuous-based sorter for higher throughput applications. Sacrifices a little bit of precision, but you can continuously sort using the exact same principles for something, again, where you maybe want to do a multi-way enrichment and do a larger throughput. Okay, let's talk about the applications, the part you're here for, right? Show me what you can do. So every single one of these examples came to us from an actual customer. So I'm going to tell you what they came and asked us to do. They said 50,000 cells. They're going to put it on a 10x single-cell RNA-seq, 90% viability, under 30 minutes, and specifically preserve the heterogeneity of our tumor. Okay, so digested tumors. And we said, okay, let's see what we can do. Enrich the viability, no problem. Bonus, dead cells never respond on our platform. Their membrane's compromised, so they don't have an electrical signature. So good news, I can go and find all the healthy, happy cells and sort those from the dead and the debris, and I can give you that 95% viability every single time. But this was the important part on this application. In equals out would be a 0% change in subpopulation, right? They knew the bead-based assay over and under selected certain subpopulations of cells, right? Just inherent in what that bead is tagging. So we monitored some of those subpopulations, and our platform kept that sample heterogeneity within 5% of its original composition versus the bead-based assay, which could vary by 20%. In addition, we did meet their other metrics. We recovered over 75% of their sample, and you can't see underneath that, but all of this was done in approximately an 18-minute sort time. Okay, so met their metrics, but it's customers, right? They want to know what else I can do. How low could I go, right? They had really precious biopsies. They were interested in possibly sequencing. 10x is getting pretty good, right? You can get down to just a few thousand cells on the sequencer and possibly still get a good map. And so they said, hey, we've got these biopsies and digested tissues, really, really small samples. Let's just see how low you can go. Okay, this is probably not realistic, but we said, let's see how low we can go. We took samples that just had a few hundred to a few thousand viable cells available, and we were able to enrich them pretty much 100% of the time. We were able to preserve those ultra low cell numbers with under 2,000 of them, and that was only in 10 minutes of sort time, small sample, right? So pretty rapid. You're probably asking me how I created live cells, right? So if there was only 450 live cells, how did I get 706? Well, as we all know, cell counting at that level is a joke. So it's mostly the cell counter that limited us, not the actual technology. But we feel really good that since we did a few thousand, we felt like we're getting most of them back, since in every single case we recovered what we expected or more. Okay, simple example in the viability spectrum, but completely different application downstream. So I'm at Virginia Tech. We're an agricultural land grant college. And there's people up the road from me that said, hey, do you know how expensive it is to breed cattle? And I said, nope, teach me. Turns out, just like humans, it's in a very, very expensive business. And the viability of the bovine sperm, when they bring it up from frozen and do inseminations and do IVF and do IUI, it's very, very low. So same thing, they came to me and said, can you do any viability enrichment? A single percentage would be great, right? Because they have no method to do so now. We can't damage the sperm. They're really, really fragile. And we need it to happen fast. Okay, done. Took the frozen sperm from 27% to over 65% in just a first pass. So that's about a 2.5-fold increase in the viability in about a 30-minute sort time. And we made sure that we didn't alter the sperm and their behavior, right? So they could keep using it again for some sort of embryogenesis or other method downstream. All right, let's take a left turn into the brain, right? Neurons, notoriously picky. They just keel over and die. Literally, sometimes they keel over and die walking them across the parking lot to us. So they're just really fragile. So this was, please just keep the neurons alive while you sort them and get them away from the satellite glial cells and debris. These are primary harvested out of a murine model. So before purification, they look like this garbled mess. After enrichment by the traditional method, they still have a lot of those garbage cells hanging around that they don't want, versus our purification of almost pure neurons on the right. But again, customers, they said, could you go a little further? Could you actually take these neurons that you enriched and sort them again? And I said, sure, why not? They're not labeled. I didn't do anything to them. I can put them back through, right? Put them back through and did small and large size trapping. To be honest, I don't even remember what the application was here. Something with the small neurons have a different trigger than the large neurons. I don't even remember, but really exciting for them because again, just because I didn't label them, I can actually pass them back through, right? And do multiple passes of sorting. And they're alive. And I note here the viability of 80%. That's what they were handed to me at, right? So I can't make up live cells, but I can surely keep them just as healthy as they were to start. This was our bread and butter. We were invented in the immuno-oncology space. Again, as I mentioned, finding those cancer stem cells from cancer cells. So just showing you here, this was from patient samples with prostate cancer, and we enriched for the CSCs or the tumor-initiating cells versus the non. And you can see by spheroid culture, we can get a 17x increase when we enrich out those cancer stem cells. And then eventually they were able to validate these with an ALDH stain. This is another one, fibroblasts and macrophages. This one makes a lot of sense, right? Morphologically pretty different. So this one's a really extreme example. You can see how fast as you watch the movie, as we turn on the parameters where the fibroblasts respond versus the macrophages, just how quickly they stop on a dime. So they're stopped here. We turn off the field, turn it back on, and bam, they all stop on those pillars that you can't see, right? They're labeled here. Doesn't mean anything to me. You can label them if you want. I don't care. It's just a visual metric, right? Otherwise, you don't believe me if I'm trapping the right ones. I get it. So same thing. I labeled these ones too. You don't believe me. T cells and B cells, fine. So I labeled them with cell trackers. It's not actually an indicator. It's just a marker. And you can see we're stacking the T cells onto the pillars here, and we're letting the B cells continue to pass through. This is a newer one for us. We're not going to get you a 99% purity right now, but we're getting a solid 80+% enrichment of either of these subpopulations, depending on what we do. So all depends what you want to do downstream with those cells. And then finally, customer came to us and said, we really want to recover bacteria from whole blood. And I said, cool, that sounds hard. And but that's okay. That's our job, right? And they said, we're really interested in the bacteria, not the blood. I don't care what you do to the blood. I don't care if it's alive. I don't care if it's dead. I want the bacteria because they're going to go downstream, try to identify that bacteria, try to understand antibiotic resistance, etc. And I said, okay. So here you can see we ran this 3 different times after optimizing our buffers, optimizing our prep protocol. And we actually recovered out of just 25 CFUs per mil of bacteria, we were getting 98% of the bacteria. So that literally means I got like 24 or 25 bacteria every single time, right? Out of this really dilute sample of blood. So pretty cool recovery here. And I just want to highlight, there was a lot of work that went into this. Oh, I don't know if you guys can see in the room, but this actually just came out in a publication last week. So you can actually see how we tackle every application on our platform. We published our process. We go through and make sure we can prep the cells onto the platform. We test our various chip geometries that I showed you, and we optimize to your application. Again, in this case, trapping and getting a really high recovery of the bacteria while I killed all the blood cells in the process. So if you think this is cool and might help you and provide you another tool in your toolbox as a flow cytometrist for some of those cell types that maybe you don't know how to tackle right now, or your customers come to you from your university and say, hey, can I please sort these neurons? And you just say, ugh, I'm so sorry, right? If it sounds interesting to you, you can of course purchase the platform. I'll tell you the price point is south of $100,000, so I'm pretty cheap for all the competitors out there. And then in terms of the rental model, we do also offer that because I am also here waiting for my funds to be released on federal grants. So I get that, and we're all sitting here stressed out about it. So we can also do a materials model. So you get the capital equipment, you commit to a year of it. All you got to do is pay me for materials. So I can write you a pretty quote that says you're only paying for supplies and you don't even have to worry about the cap expense. And you can build that into a grant 2 years from now when they renew it all, right? Just to give you an idea of some of our traction, if you wanted to go out and see what people are using the platform for, we have about to be 4 instruments in the field. I can't announce the 4th one yet, but UC Irvine, Wake Forest, and Georgia Tech are all doing completely different things with the platform: mesenchymal stem cells, red blood cells, and oncology. We have an active collaboration. I showed you the data from the immuno-oncology at the NIH. The Sperm Enrichment Project is a company that is working on improving fertility for IVF. And then I can't tell you the customer, but we're also doing age-related work. So literally any cell type, any application, we're probably trying it. I may not be able to do it all, but we'll definitely give it a try. This is our team. Greetings from my team back in Virginia. Hopefully they're on the YouTube listening like they're supposed to be, but they send their greetings. They are very jealous that I am walking around in 70-degree weather. It was 12 degrees when I left. So we send them lots of sunshine. And just for anyone in the room that works with our academic collaborators, primarily out of Virginia Tech and UVA, and then Rachael has also moved down to Georgia Tech, but greetings from our team. But I just always like to remind us, you know, I gave you lots of examples today, but I started with cancer, right? That's our bread and butter. But as you guys know, a cancer cell is not just a cancer cell, right? There's a million other cell types floating around that cancer cell. And the current estimate is that in every human body, there's 500 cell types. I'm showing you about 70 here. Now imagine disease enters the picture, right? Cancer cells, now I got sickle cells, I got all kinds of other cell types. And then guaranteed there's cell types out there we don't even know about, right? And that's what we want to do. Try to help you discover them, sort those cells, see what I can do for you. Come see me in the vendor area. I really appreciate the opportunity to present as part of the innovation session. And like I said, come see me if I can help you. Thanks, guys. I love questions. What we got? Lay it on me. How fast can I sort? Is that a question? No, they don't care? Okay, I always get the how many events per second, and then I tell you I'm not a fact, so I don't do events per second. I can sort about 100,000 cells every 15 minutes.
You could put that into seconds.
I could, but it's not an event per second. I'm not batching across that laser.
I'm kind of just curious, like as you go through with a new application, a new cell type, what is the process like to optimize that? So if somebody were to implement the platform and they want to start new applications, like is that something they can do on their own, or do they need you pretty much every time to help with that?
It really depends. So we are building a database for the cell types that we have tried. So if we have tried fibroblasts or we've tried something similar, we can probably guide you to the region of parameters that's going to be the closest, and you can probably work from there. We do offer pilots as well, where we can work more closely with you. But essentially what we would do if we're working with you to develop your workflow, again, 500 cell types. I have not tried them all yet. I'll get there if I could sleep, as mentioned. But we would take it step by step. So what we would do is make sure we know your goals, right? Is it throughput? Is it precision? Is it number of cells, right? Like, what are you looking to do? And 9 times out of 10, that's driven by what you want to do on the back end, right? So we're going to make sure we target those goals. And then again, our team would actually work to sweep all the parameters. So we have a rapid way we can sweep and characterize those parameters. And then we can build that workflow for you that becomes repeatable for you. And we do a lot of back-end validation that, again, depends on your application. So if you tell me staining is the best way to do it, if you tell me sequencing is the best way to validate, we'll do whatever validation downstream makes sense. The last thing I'll add, if there is an application where you truly have no labels, right, which does happen, the mesenchymal stem cells being a great example, if you can tell me anything about that mixture. So our collaborators at UC Irvine that now have the platform said, well, we know from PCR analysis that it's a 70-30 split. And I said, great, I'll go find a 70-30 split. And then you tell me if I got there, right? So we have ways we can work around it, and turns out it worked. You know, we got there, we validated by PCR. So we're flexible, and we'll make sure we prove the biology to you, because at the heart, that's the important part.
Cool.
Hi, it was a great presentation. And the question I have is regarding the chips, right?
Mm-hmm.
Are they like, do you reuse them, or are you able to reuse them, or do you have to create a new one for every assay? How does this work?
Yeah, good question. We don't have to create a new one, though we can customize it. We have 4 to 6 in our core portfolio that we try first. And then if we have to work from customization, we do. They're designed to be one-time use because of that sterility component. So again, everything the cells touch get thrown away. You don't have to do calibration of your laser. You don't have to sterilize. You don't have to bleach. You don't have to do any of that on the platform. You just throw it away and move on to your next experiment. I will tell you, you can try to reuse them. It is challenging though. They are flexible polymers. If you come to my table, you can hold one. They're flexible so that we don't choke the cells. And so eventually over time, they'll trap air. So eventually it'll be really challenging to actually fill your sample through because it'll become a little air vacuum. Yeah.
So, so if I'm saying like I need to sort through like 1 million cells, would I be going through 1 or 2 chips, or will just be able to do it with one? Or
Right now we only have one. We're working on parallelizing, but our continuous sorter, would be the way to go there, depending on the specificity you're looking for. We can still do, larger batches. We still try to keep anything 3 to 4 hours our max runtime.
Thank you.
And I'll just also note this one comes up a lot with the runtime. The cells are actually in the field. Everyone always asks me if I'm zapping the cells and killing them. As far as we know, no. We've done this with neurons, right, which are electrically active. We've also put cardiomyocytes on the platform. They will eventually beat in sync, which is kind of cool if you leave them on there long enough. But generally, they're only exposed to the field for a 15 to 20-minute batch time. So we haven't seen any problems preserving and retaining their behavior. Awesome. Thank you.
All right. Our next speaker is Bill Freeman. He is here to speak to us on behalf of Miltenyi. So thank you, Bill, for coming. He has been using the Miltenyi Tyto. So he's going to talk to us about sorting brain cell types and phenotypic states with the Miltenyi Tyto. Dr. Freeman is originally from Virginia, Burr, Holt, and completed his undergraduate degrees in chemistry and English. Ooh, challenging. He also earned his PhD in pharmacology from Wake Forest. Hey, maybe you should talk to Alex Hyler about that. He then went on and did some postdoctoral training in Oregon Health Science University and Yerkes National Primate Research Center at Emory. Dr. Freeman began his faculty career at Penn State in 2019, and then later moved to the Oklahoma Medical Research Foundation as a professor in the Genes and Human Disease program. He holds a research career scientist appointment with the Veterans Administration, and his lab examines epigenetic and neuroinflammatory mechanisms in brain and eye aging and neurodegeneration. Fancy. Looking forward to it.
That's where the English degree comes in real handy. Fancy words. And so for those of you who aren't familiar with Oklahoma Medical Research Foundation, OMRF, private research institute in Oklahoma City. We're across the street from University of Oklahoma and next door to the VA. So we are as to OU as Scripps is to UCSD is basically the way I describe it, just with not nearly as nice a weather. And I have to put the disclaimer that even though I'm a VA scientist, I am here actually on a vacation day from the VA, and these do not represent anything, any policies or positions of the VA. OMRF is predominantly studies a lot of autoimmune disorders, but it really has been growing the neuroscience aspects for the last several years, and we have about 50 different research groups. And so I'm not a cytometrist, but I'm going to talk to you anyway. And I'm coming from sort of the other end. I think we had really great talks this morning coming from the cytometrists and core facilities, and maybe I can provide, coming from the other direction, how we've tried to tackle this issue of isolating specific cell types from the brain. That's not, page down doesn't work, so let's try that. So what do we do? Well, as I said, we're a neuroscience group focused on aging and neurodegeneration. We look at different things in the brain as well as in the retina. And our need is to be able to isolate specific cell populations out of the brain. Yes, we do lots of single cell RNA-seq and stuff like that too, but there's a lot of stuff that we can't do in a droplet, a whole lot of stuff, especially in epigenetics, especially with DNA modifications. So we need to be able to take brains from our animal models and isolate out microglia, astrocytes, neurons, things like that. And we also do aging studies. So these are also long studies that happen over a long period of time. So we need a consistency and we need some flexibility, which I'll go into as well, that is maybe challenging to achieve just with a core-based approach. So what we've done is actually gotten a suite of instrumentation, ranging from cell preparation, relatively basic analysis, but very helpful analysis, magnetic bead-based isolation, and cartridge-based FACS. And we've actually found that depending on the study, we follow all those arrows whichever way we need to go for the particular study based on the amount of cells, the type of cells, the purity that we need for the specific study. So that looks really nice, you know, with those little biorender cartoons. Then there's the ever so unglamorous what the lab actually looks like. But part of the reason we did this and put this just within our group was really the logistical challenge, and I'll show you where we do studies that actually have hundreds of mice, where we need to do isolations and analysis of hundreds of mice. And we found all these choke points that you wouldn't necessarily think about. Like we have animals in different animal facilities within the institution. And like anybody who works with animals, you have to go clean the dirty. But then if we have to go to the dirty to get the mice, then that person can't go to the clean for the rest of the day, which makes finding that perfect 1 hour between when that person in the morning overstayed their reservation in the core and the person in the afternoon showed up early, it just wasn't working like we needed. So we were fortunate enough to be able to put all this just within our group. And we chose the cartridge-based FACS in large part because CNS cells are fragile, finicky, and the brain is full of fat, and the cells are not nice and round. They have lots of processes. We needed something that was gentle, so low pressure, We're switching between different types of cells routinely. So we want something that really requires very little maintenance, no carryover. And this is using basically disposable cartridges where we have low pressure, sterile sorting. And so for those of you who aren't familiar, it's basically this little cartridge like in the center. And then there's a little sorting window down here, which is visualized here. There we go. And basically, we've got a three laser system, and there's a little gate right here that is basically set to negative, and your cells are flowing through, and then it times it. We won't get into all the sorting metrics. That's a little slightly different thing about this type of sorting. But basically, the gate opens and sends it into the positive chamber. And so, like any sort of cell isolation, requires a good bit of optimization. But we found this was actually much simpler and suited to our needs than perhaps a conventional fax. So here's examples. I said lots of different cell types that we're interested in because a lot of what we do is we do a lot of sequencing, terabytes of sequencing a year. If you study the epigenome defines your cell type. If you have a bunch of cells mixed together, it's just a bunch of noise. You need to be able to get your specific cell types. So here's examples of Tyto sorting of microglia, where we get, again, high purity astrocytes, endothelial cells. We use a variety of cell surface markers. We have transgenic models, so we're going to have a cell type specific Cree expressing GFP. We use combinations with cell surface markers, whatever makes the best sense, and it's worked very well for us. This is a lot of stuff, but I think it speaks to what the discussions have been, and I've really enjoyed this. I've actually learned a lot already today, where we really compare and make sure that what we're getting is what we think it is, and we've really assessed all the different parameters. Here's an example where we're comparing bead-based plus sorting, Tyto sorting, conventional sorting, all of which is to say we get similar purities, but the best yields with the Tyto. And that's what I just said, actually. And this was actually really kind of an unusual thing for us. We talk about it some more, but doing two-stage isolations where we have these cell populations that are very rare in the brain, microglia, other things like that, are a couple percent. And we actually do debulking with magnetic beads and then take that positive fraction, which is often around 80, 85%, and then we sort that to get up to 95+%. That actually works quite well for us. It also reduces our sorting times, which allows us to process more mice in a day. This is the type of validations. We can then sequence these microglia in this particular case, show that we enrich for all the type of markers that we want. And then the right-hand side part of the slide here, I think, is a very important thing that was mentioned earlier, but I want to really emphasize. How you prepare your cells is critical as well. There's a longstanding sort of knowledge, but never really quite published in the field, that microglia, the very act of making your cell suspension activates the microglia, which makes sense. They're the resident macrophages, and they're going to immediately say, this is not normal to be disentangled from my niche. And so what we're seeing here is that you can do some very laborious Downs homogenization, but that depends on the person. It's a little bit inconsistent. You can do room temperature protease digestion, but it activates the cells. So we actually found that we could just introduce transcription and translation inhibitors into that cell prep buffer. And on the right-hand side, you can see here, these are all activational markers that have been defined in the field. They really don't become active like you get if you just digest. So I think that's another really important part of that before you ever get to the core, before you ever put any antibodies on it, how have you prepared your cells? So that's talking about different types of cells that we've isolated from the brain, but we actually want to go further into phenotypic states. And on the right-hand side there, you'll see the train wreck, which is the microglial field. Everybody goes out and defines their own special microglial cell about every couple months in Cell. And it's kind of a mess. And we have the belief in part that this is a little bit of an artifact of everybody relying solely on single-cell RNA-seq for defining cell populations. That data is great, but it is thin, and you aren't really looking at transcript. You're generalizing to gene and some other things. So what we wanted to do is develop a sorting strategy for at least some of these types using the Tyto of microglia. So this is again where we use, but before that we had to develop the panel. I got ahead of myself. And long story short here, we basically could use our in-house analyzer to find different populations as across aging. And this is the fun part of the slide that I like. We could define basically where homeostatic, intermediate, and these reactive or DAM microglia. And we can follow them because we took this in-house, could follow them across the lifespan in male and female mice. And so each dot there is a mouse. So that you can see homeostatic microglia in females, in males over the lifespan, the accumulation and then decline of intermediates in both males and females, and then the rise of these disease-associated microglia with advanced age. And then we can do the same thing with disease models in addition to chronological aging. And having this all in-house lets us process this many mice. That would be very hard to do otherwise. Then we developed the sorting approach to isolate these 3 different types of phenotypes of microglia. And what it does is it allows us to have sufficient material to do lots of things, like I said, that you can't just do with single cell. In your typical single cell RNA-seq experiment, you might get 1 to, if you're really lucky, 3,000 genes. We can see 10,000 different genes. And if we break that down into transcripts, because we can sort cells, do full gene length RNA sequencing, we can see about 25,000 different transcripts. We can see that they now cluster out by phenotype very well. We can find all sorts of things like senescence occurs predominantly in these old reactive microglia. But I think to drive this home, this is the next slide is what I wanted to show. By isolating cells and then being able to analyze them much more deeply, we can see things like the hallmark of senescence, which is P16. P16 is a splice variant of CDKN2A. That differential exon that you see there is on the 5' end. If you do 3' sequencing, you're going to get to about here in a traditional single-cell RNA-seq, and you do not know if you have P16, the senescence marker, or P19, the other form that's not terribly senescence-related. But because we can isolate and then sequence more deeply, we can see, in fact, yes, these reactive microglia are also expressing this senescence marker. Again, versus what we would see if we only relied on single-cell RNA sequencing, the reads trail off really at that second exon. And we can also even take these types of cells, we can isolate nucleic acids and do other things. This is nanopore sequencing for native DNA modification analysis in the top 2 panels. And we can track things like alterations in methylation and hydroxymethylation across these phenotypic states because we're sorting and not solely relying on transcript, single-cell transcript approaches, which I love, but it's not the tool that gives us everything we need. And we can also then, by isolating populations of cells, can do things like, again, nanopore, but for RNA, where we can track RNA modifications here across protein coding genes, where you see things like M6A is really peaks right there at the beginning and end of the coding sequence. So as a neuroscience lab that's been dabbling in cytometry, we are always wary of being the monkey with a gun. Hopefully we're doing well and we continue to learn. But I think for us, having capabilities in the lab, as well as when we go to the core and do larger spectral panels, it's actually worked really well from the user standpoint. And that flexibility and productivity, it lets us get so much more done versus just constantly playing a calendar game. Sample prep optimization in the CNS space is critical. And like I said, the different ways you can daisy chain these together, depending on the needs of your project, for debulking and then sorting, or straight to sorting if it's a more common cell type, has been something that we've really learned a lot about, and I think could work well for others. And I think our other experience with these they're not spectral instruments. They're simpler in many ways, which also makes it accessible to the students and the postdocs and even the more experienced staff in the lab. So that our guiding approach is that people go from mouse through analysis, through bioinformatics for their own project. We don't hand off, there's no mouse person and a flow person and a bioinformatic person. For a training environment, we want people to take the experiment the whole way through, and having more approachable instruments is helpful in that sort of training aspect. And where we go from here is using the sorting, and hopefully the folks are doing this today back in OKC, where we take the sorting and sorted cells, and then we can do actually long read single cell approaches. And then where we want to go to is where I think there's interesting possibilities in the future, where all the advances in spatial technologies, advances in cytometry, and these really should be able to talk back and forth to each other, to take that highly quantitative data from cytometry and place it in the tissue context. And so that's the group. I thank our funders. I forgot to put the OKC Thunder little symbol down there to talk a little trash, but I appreciate your attention. Hopefully that is useful, and I welcome any questions you have. I hope it is all in the paper. I don't remember off the top of my head. We found there was a feeling in the field, the reviewers, everybody who's written a grant and gets that review comment, as everybody knows, and then they don't put a reference. The thought was actually that it was the principal source of ex vivo activation was that pressure potentially. I think it's 2/3 cell prep, 1/3 how vigorously handled physically are the cells.
So I have a question. I'm not a neuroscience. We know as you age, autofluorescence can go up. And there are certain substances in the brain that are also autofluorescents outside of the normal spectra that we see, NADH and so forth. How sure are you, because you're seeing differences in age, how sure are you that it's not an autofluorescence issue that you're sorting on, like a false signal of some sort that you're sorting on autofluorescence, or versus your real target signal?
So we're compensating on that autofluorescence from the old, which gets us part of the way there. And then when we go and take the isolated cells, that autofluorescent signal, there's some in there for sure. It tracks.
And often in the neuroscience, it can be very bright and give you a very false positive signal. This is my concern here.
Yeah, and this is something we've worked on for a while. So that ultimately, I think the autofluorescence actually goes with many of these markers of reactivation. So there ultimately, total separation may not be totally achievable, but if we compensate, and when we've done spectral on these as well, we still see those shifts in those markers that I think we've done a good job of handling the autofluorescence.
Yeah, that was my follow-up is, have you gone to a spectral machine and just confirm, with the autofluorescence extraction and so forth? And
there's a couple of reports out there where folks are trying to just use the autofluorescence.
Yeah.
And what we're trying to, in a sense, we're trying to bridge the gap. You've got a few people saying that and really not really worried about cell surface markers at all. Then you have folks who are totally reliant on single cell RNA-seq. And in a sense, we're trying to bridge this to actually get past just this naming of microglial cell types and actually get towards function and regulation. I think we're doing a good job of dealing with that autofluorescent issue. It is interesting that by sorting on cell surface markers, and then confirming that with spectral flow and even just with Western blots of the sorted cells, we feel good about that. If we then take the transcript markers that many people use and look at them by spectral flow, about a third of them aren't good. And so I think the whole field, so there's three nodes there between single cell RNA-seq, autofluorescence, and what are actually good protein or cell surface markers. And hopefully we can get to that place in the middle that's reproducible and usable.
Okay, thank you.
Thanks, y'all.
We have a coffee break till 2:30, so grab some coffee. There might still be some food. So go have fun. Oh, okay. I know. All right. We're going to get started with our last couple talks of Tuesday, which is very exciting. Our next two speakers are going to do their talk jointly. We've got Rachael Sheridan and Matt Goff. They're going to do a talk entitled Think Like an Engineer, Early Detection and Troubleshooting of Cytometer Problems. Now, Rachael did do her PhD where she learned flow cytometry at the University of Wisconsin in Madison, also a very cold place. On her way here, she took a picture of the freezing tundra. So if you want to check that out, you can see it. In 2016, she secured a position at the Van Andel Institute in Grand Rapids, Michigan, where she currently serves as the director, fancy smancy Rachael there, is active in the flow cytometry community, is a member of GLIFTA's board of directors. She's the chair of the ABRF Flow Cytometry Research Group, the chair of the ISAC Flow Content Committee, an annual course director, and an ISAC SRL emerging leader, because that wasn't a lot of stuff. Her cytometry interests include instrument characterization, cell sorting, non-immunological, wow, this English degree is really failing me, assays and methods to support single-cell genomic methods. She plans to continue building cross-core collaborations and asking what if. Beyond cytometry, she enjoys being active. If anyone wants to go for a run with Rachael in the morning, she is currently looking for people to run with her in the morning. At a slow pace, she encourages those of us not active. She also plays with her kids and she eats food. Lots of snacks. Also, if you're ever looking for Rachael, she is active on the Flow Cytometry Discord, especially at 9 p.m. So if you have any data questions at 9 p.m., know that Rachael will answer them. I will not. Matt, on the other hand, began his flow cytometry journey back in 2005 while he was investigating environmental toxicology and its effects on developing immune systems. Interesting. From there, he has been a research associate and a core lab manager before moving to industry. He now works for Beckman, in case you guys didn't know that. He is leading the flow cytometry hardware team there and focuses on new product innovation. But in his spare time, he does do fun things, believe it or not. He likes to go camping with his son, especially where cell phones don't work, and he cannot respond to the Discord at 9 p.m. at night. He also spends time with his family and friends all around the world. Any specific places? Anywhere fun? He spends a lot of time in China. All right, have fun.
Always the hardest part of this is figuring out how to advance the slide. So let me get a test run real quick. That's not the key. Which one is it? Click. Which click? Any click? All right. Technical challenges have been abated. All right. So the title of the joint talk, Rachael and I decided to go on tour. We did this talk first at Cyto last year in Edinburgh. It was a lot of fun, and we're pleased to bring it back to you again, hopefully at least fractionally improved. The title of our talk today is Think Like an Engineer, Early Detection and Troubleshooting of Cytometer Problems. Everyone can read the slides, so I'll tell you the summary here that we want you to focus on is the concepts we want to talk about are instrument management. So things that you need to be thinking about in terms of the life cycle of an instrument and how it's going to be managed both for care and for use when it's in your facility, and what your responsibilities are based upon the role that you have within that responsibility. We want to challenge some paradigms that we see that exist, that we've detected through conversations, educational events, and a variety of other sources, and get you thinking sort of, I dare say, outside the box. It's a bit of a hardware joke, haha. And so looking at what other information your flow cytometer can give you and how you can interact with it to basically have a better relationship. with your flow cytometer. So with that, I have to do the conflicts of interest and disclaimer. I'm employed by Beckman Coulter Life Sciences, and I'm responsible for product associated decisions, including creation of marketing and new and existing products. That said, the opinions in this presentation are my own and do not in any way reflect the opinions of Beckman Coulter Life Sciences. And any data shown or products discussed, regardless of the instrument or application vendor, are representative examples and should not be seen as an endorsement. So with that, we'll start, and let's just jump right into it. The first thing I would tell you about any instrument you own is, and I don't know how many of you are old enough to have played Portal, but thank you. Yeah, there's a few of us nerds around. Is that the QC is a lie, sort of. And conceptually, we're going to work our way through in my part of the presentation what I mean by the QC is a lie. Okay. We need to talk a little bit about how we get to QC and what are some of the physical characteristics of the beads that we use, because almost invariably we're all using beads for the built-in OEM QC. And we need to talk then as a result of that about what your QC is, and then more importantly, perhaps what your QC isn't. And then conceptually, if you've never heard the words before, and this is certainly targeted at those of you that haven't been doing this for 20 some years, I want to talk to you about the concepts of performance qualification and characterization. And these are terms that are actually pretty popular in the manufacturing world for these instruments and in manufacturing in general. So I want to touch on some topics that we use as manufacturers and how we describe and build instruments. Okay. So the first thing that we should discuss is hard dyed bead behavior. So most, if not all, of our QC apparatus is based upon hard dyed beads. Okay, and so polystyrene beads are typically the most popular from a variety of manufacturers. These are generally polystyrene beads that are either stained or polymerized with a fluorophore. Now, those fluorophores have interesting names like light yellow, Nile red, pink, purple. Quite literally, these are the names of the dyes that are used in some of these. And sometimes you have other stains. There's I believe there's Kumasi and Coumadins, and there's a few other different classes of dyes that exist. But we've reduced this to a functional practice on a commercial level. And what we're able to do with these dyes, as you can see in the picture on the right, and this is the same microscopic image of the same beads in a field of view, is that they can exhibit different characteristics on the same dye using, or on the same beads using different dyes that are all polymerized to the same bead. And so this is how we get what we call a multifluorescent particle. And so when we shine, and I'm going to use names rather than wavelengths just for brevity, but if we shine blue laser light versus yellow or yellow-green laser light versus violet or versus a red or a near infrared or a far red, we can get different resulting downstream, right? So it still follows the rules of Stokes shift. We can get different downstream behaviors with these dyes. Now, what these dyes really represent for us versus using a non-bead particle, what these stained beads represent for us is a couple of behaviors and limits. And the first behavior that we really like about these beads is that they're consistent. Now, we all have, this is the part where I have to say, I'm not going to sound like Beckman Coulter for a second. We all have beads within our reach that are probably well past their expiration. Actually, this will be a fun test. Raise your hand if you know that you have beads in your facility that are past their expiration. Keep your hand up if you still use those beads on a regular basis. Right. Thank you. So what we know is, first off, is that as manufacturers, we have a responsibility to say we guarantee the performance of this product up until a certain point in time. But I can tell you also anecdotally that we regularly put hard stained beads in our pockets and fly around exposed to cosmic radiation on the regular. And in general, they work just fine. They're very, very robust. They're very consistent. They're very resilient to environmental conditions, including temperature changes, with one exception, right? And who knows what the exception is? Freezing. They don't like freezing. Okay, so that is the one thing that they don't really like. But we've seen them survive some pretty high temperatures and some pretty low temperatures. But freezing on the balance is not great for your beads. The other thing that your beads can give you, especially if you characterize this quality, if it's not guaranteed by the manufacturer of the beads, is concentration, right? So that's a piece of information that you can actually calculate in a lot specific fashion or in a bottle specific fashion, and you can use that information as part of your determination. And I say this about QC beads, but really it's true for any polymer bead, right? If you uniformly resuspend them and you distribute them through a consistent process, you can maintain the concentration of these from the first drop to the last. And so that's a useful characteristic of beads that can really help you understand what the running conditions of your system are, especially when you've got nominal pressures being applied, either through positive pressure or vacuum to your sample. Okay. The other thing is, and this is getting just like an order of magnitude beyond our QC beads, but polystyrene beads are also great because we can anneal functional groups. And so we can do some interesting things with this. We know that there are such a thing as capture beads for when we want to capture antibodies and we want to do different kinds of, library building and staining, compensation or unmixing, groundwork. And so they're very, very diverse, right, in what they can provide for us. But now I think that the thing that we have to talk about is their limits, okay? When not handled properly or when stored for excessive periods of time, they can aggregate. And the longer that the beads have sat, the more that they will aggregate. And so if you're running beads from a bottle every single day or every other day or every third day or even once a week, likely you're going to have limited aggregation. But if you're using a specialized instrument more infrequently and your bottle is sitting, or you're mixing bottles and you've got 2 or 3 open bottles, or you've got labs that are bringing their own bottles to do their own QC, right? All of these things can confound the behavior of the beads. And we're very, very quick to say there's a problem with the instrument. And so the point of this is to say, you have to know what you're running before you can, jump to the conclusion that your instrument has a problem. It may be a reflection of what your expectations are that can't be met because the beads are not actually in a format that they should be using. So you have to pay attention to how they've been treated. And this is one of those cases where I say the role of the flow core, which is what a lot of the people in this room are associated with, is not just to maintain the instruments, but also maintain the things by which you're judging and determining the quality of your instruments. And so I do think it's important for us to take on responsibility and say, think about like, how have you treated your beads, right? And to have substitute products ready to go, because that can oftentimes short circuit what could become a major, major disproportionately determined, blown out problem. The other thing that we have to remember is this, and this is something that I think is easy to lose sight of, even if you've been doing this for a long time, and that's you have to stay grounded in the idea that the dye that's on these beads is not equal to the fluorochromes that we have available to us. And you notice that I used a different word, fluorochrome versus fluorophore, right? So the fluorochromes that we have available to us as either derivatives of a natural process or a polymerization process that are attached to our antibodies, or I guess we also have some nucleic acid targets now. Those are fun to play with. But the fluorochromes that are associated with those are not equal to these dyes, right? They may fluoresce in the same, in the same filter set. They may have similar emission characteristics on some limited level. But especially as we transition more and more into a spectral world, we have to be cognizant of the differences. And this is also true for the thing that we don't always talk about, and that's molecular fluorescence. And we could even go a step further and remind everybody that if you're working with things that have, very, very broad emission patterns, 7AAD, right, PI, DAPIs, that we also have to be cognizant that the fluorophores that are associated with these beads are not equal to those either. So be aware of what the representative limits are of these dyes, and be very careful what conclusions you do and don't make based upon the performance of those fluorophores. The other thing that we have to take into account is autofluorescence. Not necessarily the more or the less elements, but that it exists and that it's different, right? Most instruments in this day and age, and I have to say most because what I really want to say is I can't think of an instrument, but I'll say most instruments in this day and age have the ability, when you're setting up a library, whether it's for traditional bandpass-based compensation or it's unmixing, to identify what you're using for your single colors. And you'll often, if not always, given the option to say whether or not it's a bead or a cell. And a sufficiently sophisticated instrument should be able to manage the autofluorescence contribution from a bead versus a cell. Now, I also realize, having seen some wonderful talks, both here and in other conferences, that we are starting to really hone in on the autofluorescence question. And we have differentiated population-based autofluorescence in addition to different states of activation, inactivation, cell life, death, right, everything else. So I think that this is going to continue to be a very interesting question to see how all of the manufacturers take into account the varied states of autofluorescence. I love the question that was asked in the panel earlier, and I'll paraphrase it for those of you that weren't watching, and that was, how are we managing autofluorescence in these in-by-ins? How are we managing it with the graphic works that Dr. Page showed us? It's really a very interesting paradigm. As we can see more data, we have to decide what we want to do with that data. So the autofluorescence thing is very exciting to see us continue to evolve in. So an example of a QC report, you can see one on the left. This is from My Aurora. Thank you, Rachel, for these reports. And you can see some of the things that we pick up on the QC. And so you see the name of the detector, the gain value, which is the amplification that's applied to the detector in order to get the resultant signal, what the gain change was. This is our first delta value, so I'll refer to this as a delta. This is how much the gain changed from one day to the next, or from one QC to the next. It doesn't necessarily have to be a single day. There's, your median values. This is going to be your MFI, right? These are in arbitrary units. And then your RCV, your percentage of robust CV. And then the instrument is typically going to give you some kind of a yes or no. Does the channel pass based upon the boundary conditions that are applied to the QC report? On the right, we see a similar but different report coming from a Fact Symphony S6. And you see, again, the same basic things: target value, actual value. So some of the data is truncated in the previous report, and that's entirely okay. Percent difference target value, this is rounded down, right? This is opposed to a gain change value. It's just shown as a percentage. But this is actually from CS&T, right? And so if you're familiar with CS&T, this is an enhanced version of QC offered by BD. And you can see that the bright bead, mid bead, and the other mid bead characteristic are called out here, right? So we get this data. The trick is, what does this data mean? What can we do with this data, right? And so that's the real question about how much does our QC influence our belief that the instrument is operational, and how is it suitable for our use? And they're very different things. And so that lets us dive into what is QC, right? In its strictest terms, QC is a representative from the manufacturer of what we call instrument qualification on an ongoing basis. And so what this means is that the instrument is operational and it is qualified to begin an experiment or use, meaning that whatever we represent on a spec sheet has been accomplished through and measured and provided and described through the QC process, right? But most importantly is that QC is a measurement of consistency. How is the instrument operating day to day? Almost all instruments that I'm aware of have boundaries and delta value conditions that are built into the QC that describe and control the tolerances between one day to the next. And so while the instrument may be able to make the appropriate adjustments based upon barometric pressure, overall volume of fluid in your tanks, any of a number of external factors, it may be able to make the adjustments needed for things like laser delay, timing, some of the things that are even more cutting edge out there, and some prototype instruments are beam spot location, right? There's all kinds of crazy stuff that's available. What you're really trying to do for the user is to keep the instrument consistent from measurement point to measurement point. Oftentimes it's day to day, but it could be obviously longer than that. And so to do that, we have a number of specifications. And so you see these listed, right? So laser power and delay are here. This is one of the early critical factors, right? Can the instrument at the point of interrogation deliver a consistent shape beam that also has consistent power, right? And there's a lot of different ways that manufacturers handle this. Some of us use a Gaussian beam shape. Some of us use what's called a flat top or a hat top. And all of them have their place and all of them have their time. What's important, though, is that you understand how your instrument's operating in this space, right? So if you're used to seeing a delay of exactly 20 microseconds or 30 microseconds, and you see a slip, right? What does that actually mean? And Rachel's going to get into some of this, overcoming these challenges. What do they mean? What you can do about them? But laser power and delay tell us something about the nature of the interrogation that we have and how consistent it is. But delay tells us, in fact, it's really probably the best measurement indirectly we have of temperature, right? So we could fall down a very deep rabbit hole in this, but temperature is probably the thing that we manage sometimes the least, that has the biggest effect on our flow cytometers. And I think anybody here that's managing a sorter knows especially what I'm talking about, because sorters tend to be even more sensitive to changes in temperature. So this is one of those things where if you see changes in delay, right, we're going to start thinking about environmental conditions, the temperature of the sheet. Did you just change the sheet, right? Is it degassed or not degassed? Has there been a pressure change in the room? Has there been a temperature change? Are you doing your QC at 8 a.m.? Or are you doing your QCs at 3 p.m.? Right, morning drift is a real thing. And I think we've probably talked about that over the years. The next thing is the robust CV, right? And this is typically going to be a measurement of a gated condition. And so if your multifluorescent bead is emitting in a particular channel, there's a width to that emission. Now, the brighter the bead, typically the lower the RCV is. But there is such a thing as having a bead that's too bright, right? You want to measure the conditions of the flow cytometer relatively close to the abundance of fluorescence, and I'm talking about traditional bandpass-based architecture, that you can convey with this fluorophore that you would get from a fluorochrome or from molecular fluorescence. Now, obviously, there's some interesting challenges in that. When you are dealing with a channel that measures both, say, FITC and GFP, how many of us have encountered a GFP that's completely off scale and is entirely cannot be represented by any QC value, right? It pegs your MFI, 5, 10, 15 million MFI based upon the settings that you would traditionally use for FITC. So we do still have challenges here in terms of the representation. But in general, you want to have a bead that's bright enough to maintain a consistent width of peak. That RCV is typically going to be a spec that's called out by the manufacturer. And if it's not called out on a sheet, then it's going to be programmed in, because there's an acceptable tolerance and there's an acceptable delta. There's an acceptable change from one to the next. And then there's also delta values. So how much change over time, not just nominal values, are you willing to accommodate? So that comes in. Then the last one here that we see that's also tied back to temperature and delay is going to be the event rate. And so mostly, even if the spec is only controlled within 10 or 15 or 20% in terms of the concentration, if you were to say take a set of beads that have a nominal concentration of 1 million particles per ml when they're properly resuspended, the instrument is programmed based upon those instructions. So you followed your IFU or your user manual, you've resuspended your beads correctly, or you've diluted them properly according to the manufacturer's instructions. And then you're running them, and then the instrument should know that you need a minimum number of events per second in order for the instrument to convey that it's operating properly, right? It goes back to instrument qualification. But you also don't want to be so high, right? So, for example, you may want to be at a minimum of 80 events per second, but you don't want to be above 1,000 events per second. So 2 orders of magnitude or 1.5 orders of magnitude are basically contained in there so that you can have some range in the resuspension. What is measured is going to vary based upon the bead. So we have basic, single point QC that's measured, right? Typically, it's going to be 1 color per channel, kind of a thing, single bead. And then you have more sophisticated versions. Obviously, CS&T exists for this purpose to do things like, like area scaling factors and more sophisticated multi-size delay. What's reported also becomes important. And so what you can see on the screen and what you can output, think about what your purpose is here. Do you have a need for something like 21 CFR Part 11? Are you in a either compulsory or voluntarily regulated environment where you need to have chain of custody on your QC as part of your laboratory report so that you can say, when the instrument was QC'd, before we did any sort of assay or experiment validation, we verified that the instrument was operating properly. And so that becomes a question too. But remember that with instrumentation, and if you're in this world 21 CFR, it's always tool sets, right? There's no automatic way to be 21 CFR Part 11 compliant. It's a behavior, it's a choice. It's never something that's automatic. So can you be 21 CFR Part 11 compliant without built-in tool sets? It's entirely possible, but you have to talk yourself through the process and make sure that you have the process validation that's going to create that, okay? The real question that we get into then is where we started, right? So if I say the QC is a lie, what am I saying it's lying about? Well, we can ask QC a lot of questions. And we know that these questions we ask of QC all the time, right? The consistency and the delta delta and the other boundaries that are maintained. But what QC can't be, and the mistake that we've seen made over time, is it can't be an adequate representation of performance qualification. And I separate this in terms of an idea from instrument qualification, right? So performance qualification has to do with what you want to run on the instrument, okay? And validating that it can be run the right way. So you still may not want to use your valuable experimental samples or limited experimental samples in order to do this. This is where we start having to think about instrument management beyond QC. What are the other proxy particles or materials or methods that you can use in order to feel confident that a qualified instrument that's running the way that the manufacturer describes it is also operating in a valid fashion for you to do your assay, right? And so what we talk about here are steps that you might take on either a regular or some recurring basis around determining your instrument's operating state beyond the consistency. And so one of them would be antibody titration. As we expand our panels, as we deal with our reliance on, just lost the word, forgive me. As we expand our reliance on more complicated fluorochromes, as we expand our reliance on these donor relationships that break down over time, right? Knowing how your antibodies are behaving over time becomes critical. We've seen things fall apart in a tube. We've been able to measure where we're actually getting more fluorescence from the secondary fluorochrome than we are from the primary fluorochrome. You can trace this over time. So I would say that antibody titration and quality checks become really, really critically important. You don't want to make the assumption that your instrument is not measuring something right when actually it's just the antibody itself that's degraded in the tube over time, in spite of handling. How many of you have antibodies that you're not the only one who touches them? Okay, so do we always know that they were perfectly handled the way that we would handle them? Okay, are we capable of making mistakes? How many of us have ever put them in an ice bucket and come back and said, that ice bucket's still on the counter after lunch? And you're like, oh no, right? And we walk and you lift the lid slowly and you're like, oh good, there's still ice, right? You rush them. So the point is that the broader and bigger these panels get, sometimes the less we're even using of these antibodies, especially when we're not reaching into the fridge for the things that are giving us these answers that are an order of magnitude beyond sort of our core panel, right? These drop-ins that we might use through different strategies. So it's important that we know what the performance of those antibodies is in relation to the machine that we want to use them on. The second is voltage or gain tration. And it's clunky to try and say it both ways because really it's just looking at the voltration of an instrument, whether it's called voltage or gain. And so in this scenario, we expect that modern instruments built with mostly solid state silica components maintain day-to-day and even, I would argue, year-to-year performance. And for the most part, they do, right? The instruments are far more consistent and more likely to show an error through a QC process before we would even detect this problem. But it's important to understand, especially if you are working with libraries, that you really, really need to have done this voltage gain tration. If this is not a service or a procedure that's offered by your manufacturer, this is something that is well within your power. It's well described by a number of papers, and actually we'll flash them up here in the resources at the end. This is something that anybody can do with the appropriate beads, right? It's very straightforward. But understanding not just where your linearity exists, but what the range of linearity is on your system has a huge effect on the limits that you can apply to your library, right? So we know the library values can be confounded by antibody degradation over time, especially with tandems. That was the word I was looking for earlier, and I don't know why I forgot it. Forgive me. And we also know that that library values can be confounded by changes in the linearity of the detectors, right? So it's important that if you're relying on those things to make conclusions about your data, that you're also monitoring them and that you have a Sentry program to maintain those things. We often overlook this responsibility. Why? Because it's a lot of work, right? But is it work you have to do every day? No. It's work that you can probably do once a week. It's probably work that you can do once a month. But it is work that you should schedule in as part of the active maintenance of your instrument beyond just deep cleans. These things are going to give us determinants of quality quality interrogation, right? And again, they're cost effective for a lot of reasons. The investment that you're going to put into the particles that you would use in order to make these determinations is going to save you lost assays, especially as we move into more and more complicated things. You do not want to find out that the assay settings that you used, which worked perfectly well 2 or 3 weeks ago, now suddenly have a problem because of a degraded tandem or because of a changed setting that's required in a detector to bring it back into equivalent operation or comparable operation. So there's some other things that you can do too with this work, and it's going to give you the linearity determination, the dynamic range, and then also it gives you this nice thing that we call the sensitivity measurement, right, which is QBS, if you've heard of that. So we don't have time to get deep into the papers here, but the way that these have been written by cytometry experts, they're infinitely followable. They're very, very simple and straightforward. And there's a lot of people in this room that would love to help you if you're having any trouble with them. So with that in mind, I would also introduce you not just to single peak beads, but other things that you could consider as reagents. Now, you can go old school, and if I make portal jokes, then I have to talk about CEN and CTN, because that's how we used to do things. So with chicken erythrocyte nuclei, you can see on the right, this is what we used originally for fluorescence linearity. It still has value in determination of instrument state, so that can be very, very useful. And then on the other side of this, we also have the calf thymocyte nuclei, which has some of the same characteristics as the multinucleated chicken erythrocytes, but also gives us the advantage of being able to do double discrimination. And these are taken from BioSure, which is still an active provider, is where I got these images from. So if you're interested in using these old school methods, they do still exist, and they're totally accessible. You also want to consider the use of 8-peak and 6-peak beads. This is where we get into what we just showed in the other slide. So QBS type questions, linearity questions. The basic fundamental truth about these beads is that the staining pattern on them is linear one to the next, and that you can use them as a representative model. I like to trust things that are tested rather than trust them because the paper says I should trust them. So I always try to, have to minimize my variables. So even something coming out of a bottle, I like to test and make sure that it's what it says it is. If you're interested in doing things associated with scatter properties, especially as we see, different versions of scatter based on either wavelength or angle available, this is also something to consider. Size and scatter are not linear one to the other, and I'm really happy that that was already said once in this Congress, because it is a lovely fallacy that gets contributed to by misunderstanding and perpetuated through time. To put the Beckman hat on for a second, as somebody that has worked on the release of a nanoscale particle instrument recently, I can tell you that the lessons in scatter are deep, and they get very deep very, very quick, right, when you get into like me theory and stuff. But super fun if you're interested in those kind of things. The other thing that I would recommend that you consider too is MESF and ERF beads and ABC. So there's a couple of different ways that we can work towards truly quantifying our fluorescence. The beads that we get that are classically referred to as 8-peak or 6-peak beads are actually MEF beads and not MESF beads. MESF is the designation that we reserve for a true fluorochrome like FITC or PE or APC, directly and chemically appended to a series of beads in a concentration-dependent fashion. There are other theories out there that can give you this information. NIST has the ERF effort that's well described by the NIST Flow Cytometry Consortium, and then you can obviously do this through a DIY method using antibody capture particles that are sometimes referred to as ABC, at least if you read my emails. What this is going to give us is more advanced information on instrument characterization. And you can see on the left here where we do a gain or filtration walk. And what we're looking for here is to push the signal up to the point that we're pushing the noise up. It's really actually quite simple. Once we're pushing the noise, once we are amplifying the noise past a certain point, we've gone too far. And what we want to do is we want to capture as much dynamic range as possible. You may also have analyte-specific needs and for dynamic range. We talked about GFP and how some of the synthetic GFPs are incredibly bright, even in low quantity, right? And you can get into some very specialized instrument management tasks when you're dealing with certain things like that, the introduction of additional filters, right? So there's all kinds of fun there. And then on the right, you see this is coming from that chicken erythrocyte nuclei, how we're using haploid, diploid, and different aneuploid states in order to do linearity determination outside of a hard stain bead product that is, assembled by a human. So we would use a natural biological process where we're staining the nuclei versus a hard stain bead. So that's a good way to check them one against the other. And so with that, we'll summarize and hand it over to Rachael. The questions for determination of instrument performance, when we look at quality control, we're asking, is my instrument performing within the designated tolerances as described by the manufacturer? And has my instrument changed more significantly than is tolerable since it was last considered within those acceptable tolerances? That's a lot of words for saying, is the instrument basically running the same day to day or from checkpoint to checkpoint? Separately from that, we talked about the performance qualification, which is, does my instrument operate in a way that allow me to measure the parameters I'm interested in with confidence, and how have I checked that? Do I know the limits of my instrument's interrogation, especially associated with dynamic range linearity and resolution? And have I tested those? And is my instrument capable of creating data that can be compared to results from other instruments? And this is a very interesting paradigm, because the idea that you have to have all the same instrument is, I think, a little bit crazy. I think that we should be able to have procedures and policies in place that allow us to have comparable sets of data. And I'm proud to see the field slowly, glacially moving towards that, but more progress in the last couple of years than in the previous 40 for sure. And then from assay settings, right, this is, have I selected the appropriate settings to interrogate my samples? And we talked a couple times about the wild cards and the outliers and how you would validate this. I'm flashing this up so it's on the screen and you have it in the recording. These are some of the resources that I recommend that are helpful for understanding some of the concepts in my talk here. And with that, I will hand it off to the glorious Rachael. Thank you.
Spoiling my thunder, Matt. So Matt talked about a lot of things that are best practices and good ways to set up your instrument and determine how it's working. I'm going to shift gears a little bit and we're going to move into the lab and what you might find on any given day. So, we may or may not have service contracts, but we take care of our instruments. We do QC and they're going to be beautiful. And maybe it is in the morning, but how are you going to do? Right? Yesterday's performance is not predictive of today's. And as my lab is currently in the middle of dealing with, this morning's performance is not necessarily predictive of this evening's. We had an instrument basically lose a major electronic component in the middle of a run. Things happen. Sometimes there are things you can control that you can deal with. Sometimes they're gremlins. So as Matt mentioned, temperature is something that can influence our instruments. Some of the things we can control, cleanliness, we can check for leaks. If we see salt buildup, we can be proactive about that. Sheath tank levels can change how your sorters behaved. And full characterization isn't a feasible daily operation. This is a thing that you can do once at the beginning and maybe periodically. But for day-to-day, you're kind of reliant on a more simplified QC and characterization. There will be some specific instruments and some specific QC reports in this talk. They are not meant to pick on anything. They are just either the prettiest pictures or the QC reports I had closest to hand. I have 4 instruments from 4 different vendors. I love them all for different reasons. So evaluating your daily performance. So you've got the manufacturer QC that Matt talked a lot about. So we look at things like the bright bead CVs. If I look at beads yesterday and today, do they look the same? Do I need the same voltage settings to put them at the same place on the plot, et cetera, and so forth? Not all instruments will give you QR and BR values in their QC report. Not all instruments will show you an area scaling factor and or track it. And this can be a thing that's important if you're going to go through and characterize your instruments. If you don't have access to an area scaling factor and you are trying to characterize everything on area, you may be getting area values when your height's already off scale and running into problems. So I do recommend checking height and area for whatever you're running if you don't have area scaling to look. In-house QC, a multi-peak beads. I'm used to using 8 peaks, but I think the ones that work better off the UV are only 6. That's fine. In the sort world, there are other parameters that I like to track that are not tracked within the software. I like to track these sort settings, things like amplitude, frequency, the drop one, drop delay, assay-specific tracking as well. I like to have process controls. If I'm going to be running samples, the same panel over time, if it's something that I can get a process control for, like PBMCs, I'd love to buy a big old batch of cryopreserved PBMCs and run one aliquot every time I run that assay so that I see if I did something wrong. It's also nice if you want to go and use some of the normalization algorithms because it gives it something to grasp onto. Not all samples are amenable to banking like this, but if you can find something you can bank and run along with your test samples, it can save your bacon later. DNA QC controls, I actually use CENs and CTNs outside of instrument characterization. I run them all the time when someone says your instrument's not running my cell cycle right. Just throw on some of those guys, and if you see your beautiful peaks, you're in good shape. So how do we visualize these? It's hard. You don't remember what the value was yesterday, 2 days ago, the last lot of beads. And so this is where our Levy-Jennings plots come in. Many cytometers software has these built in. If not, you can export the data and make them in Excel. So the plot structure is usually date along the X, and then the value of the Y is whatever it is that you're monitoring. It's a plot that looks over time. And so you've got kind of like the average, and then you'll usually have dashed lines for 1, 2, 3 standard deviations, whatever you're okay with. Pass-fail conditions in your QC software are arbitrary. They're whatever you and or the manufacturer decided to set them to. So you need to decide if they work for you. Some software allows you to update them so that you get pass-fails that actually make sense. Another thing that you may want to have trigger a pass-fail in your brain or your team's brain is a certain number of values above or below your average. That could mean that something is starting to go but hasn't quite triggered a full fail yet. So if you routinely see a smaller change, it may be something to think about. So you've taken care of your instrument, you've characterized it, you're running your QCs every day, but one day it's gonna happen. Your QC is gonna fail, or worse, the whole instrument's gonna fail. But so now what? I talked to one of my favorite FSEs. He's been in the field a really, really long time. And this was actually his first piece of advice. He said, don't panic. Take a couple of deep breaths. I thought rainbow breathing would be appropriate for this field. So that's the one I've got. If you prefer another one, go for it. It's hard for us to think when we're in panic mode because our user's gonna be here in 30 minutes. Now, what are we gonna do? The second piece of advice he had is also commonly heard in medicine, but if you hear hoofbeats, think horses, not zebras. We are shockingly close to a zoo here, but I still think the odds of finding a horse are a little bit higher than a zebra, unless there's been an escape. Okay, so when you have a list and you're like, these are the 3 different things I can think of that can cause that, think of the simplest and most likely one to happen and test it first. If there's a really hard thing to test and a really easy thing to test, test the easy thing to test first. Just work through it and eliminate what you can. I tend to take a very expedient and practical approach. If you are clinical or in a highly regulated environment, please plug your ears. So what actually failed? That's the first thing. If my team says, hey, this instrument just failed QC, I'm like, how did it fail? What value is triggering? Did it fail in a good way or a bad way? Did we have a detector just get more sensitive? If so, I'm kind of okay running with that and I'll chase it down later. If we got less sensitive, I might have to think about that. Did the failure occur on a channel or a laser or an array that you need? So your red failed, but you don't have to run any panels with any red dyes in them. Or you've got APC, but it'll run off the yellow-green okay, and you can limp through. Is there a secondary channel you could use? Do you have time for you or the user to change the panel to avoid the problem region? If it's an issue of, say it's a sorter and you can't do drop delay because for some reason, the optical filter decided that it just wasn't going to software. Is there a manual way to do it? Or can you pull one of those historical values from that spreadsheet that I said you might want to consider making and then validate it? And so again, and do your facility culture and regulatory constraints allow you to do this? So if you can find a way around the problem to keep going for the day, 2 thumbs up, block it for when you can have some time to breathe and focus on the problem. If you're lucky, QC will even give you a helpful message. Sometimes I get the event rate too low. Usually I know manufacturers give us instructions on how much beads and how much sheath. I use a squirt bottle and I guess. Sometimes I guess wrong. Or they were sitting there too long and I didn't vortex them well enough. So double check how you make your tube. That's a really easy to deal with error message. Can't put beads on scale. Kind of don't like this one because that can be a number of things. So this one is just like a quick list. Like, are my lasers on? Because sometimes I forget the coherent connect exists. Did I even load beads or did I grab the tube of water? Right? Do you have an ND filter where maybe you shouldn't? Are there any weird thresholds where you're running an unusual assay and so now you've accidentally thresholded out your beads when it started looking for them? And so one of the things that I like to do when I get these kinds of error messages is just open up a new experiment outside of the automated QC routine and load my beads. Can I find them? Do they look pretty reasonable? Okay, let's try it again. If they don't look reasonable there, then we have to do some more troubleshooting. And once we start thinking about the additional troubleshooting and thinking through it, we have to realize that our QC metrics are not just impacted by one thing, right? All of these values that we're tracking can be impacted by multiple variables. So our detector settings, our CVs, delays, all of these can be impacted by fluidics, optical alignment, optical cleanliness, our various reagent characteristics. So there's not always one clear path to go down. So I like to build mental models in my head. And I don't think that this talk today is going to tell you how to fix everything you've ever seen, but I'm hoping it'll give you a foundation and a thought process for when you run into something. So we are all about balancing a whole number of different forces. Think about the journey that your cell goes on. So it starts in the sample tube. It has to somehow move through this whole sample line, go through hydrodynamic focusing, go through sample illumination, collect those photons, and process all of those photons into electrons and through the electronics and data to the software. And if you're using a sorter, you've got all the sort functions too. And even within this, within the sample illumination and photon collection, we've got our core stream. We've got to get from laser to laser to laser through the pinholes. We've got the forward scatter module with the obscuration bar. So there's a whole lot going on in here. And all of these have to align just right for us to get good quality data. So we've got to go hunting. So what we're going to start with is looking for other clues in the QC. So sometimes it has an error message that's helpful, but sometimes it's just a failure and you've got to think through it. So some of the things I look for is, was it just a single channel? Was it a whole array? Was that single channel forward scatter? Because forward scatter is on its own little detector unit. Are all of the arrays affected? Was this a sudden failure, or has this been kind of coming for a while when we look at those LJ plots? Okay. So single channel fail. This is where it gets interactive and I get to pester you guys. What would you check first? If you see one channel fail, who thinks it's going to be fluidics? Laser alignment? The post-excitation optics? Photon detector, your PMT, your APD, whatever you want to use. Or your bead reagents having problems. Man, not too many brave folks. So in this case, when it is a single channel within all of them, I lean towards checking my optics, my post-excitation optics first, right? Fluidics tends to have a broader effect. Laser alignment tends to affect that whole array. Bead reagents also tend to have a more broad pattern. I'll get to the photon detector in a sec. So this is 2 examples. This is off my sorter. One day we failed because we got 90 volts more sensitive on forward scatter. And then another day we had this failure on violet A. The The PMTV delta was up 55. My team tried cleaning. We did all sorts of things, and it just wasn't moving. And then I remembered that that pesky director had gotten her hands on an instrument for some reason and taken out a filter to look at something and may have put a fingerprint on it accidentally. So we cleaned the optic and got it back. And the forward scatter fail was, we ran CS&T with a different ND filter in there than CS&T was looking for. So I figured that out. I'm probably not going to bother rerunning CS&T if I'm short for time. So in this particular case, as these were both consequences of my own actions, but these single channel fails tend to be something more like ND filters and dirty optics. It's quick and easy to clean those and check those, so that's where I start. So another one in that list that people may have been thinking was, oh, that photon detector is going bad. They can. It doesn't happen often. So, and they're a lot harder to troubleshoot unless you've got spare ones around and are really comfortable rerouting the cables in your instrument. Yeah. So I would rule out everything else before I start looking at your APDs, your PMTs, and unless you're super comfortable in there, I'd probably call your friendliest engineer. So what if an entire array is failing? Say none of your violet's working or none of your yellow green, everything is failing, the CVs are too high. Who would look at fluidics? Laser alignment? Post-excitation optics? Photon detector? Bead reagents? Okay, so most people were thinking fluidics or laser alignment. Probably a good choice. And so here's where I want you to think about which one is it? Your laser order matters. So these laser delays are always relative to the trigger laser. Standard in our field is the blue. Doesn't have to be, you can change it. So if you have any fluidic deviations, say something is there's a partial blockage and now your stream is running just a little bit slower than it was. The laser furthest away from your trigger laser is going to be impacted first. On my Cytoflex, that's the yellow-green. So if I see the yellow-green starting to have problems and everybody else looks okay, I'm going to do a deep clean and hope that it's a blockage I can pretty quick clear. Can you manually recover signal by changing the laser delay? So if your yellow-green is the one that's starting to look a little crummy, if you make that the trigger laser, does it look fine now and a different one is failing? So if you can shift that fail around by changing your trigger laser, it's probably a fluidics issue and you've got bad laser delays. In this case, soak clean, debubble, soak clean, debubble. Cleaning is the answer to a lot. If you've never manually adjusted laser delays, it's a really good thing to know how to do, especially if you have sorters. So you choose a bead, needs to fluoresce anywhere, so pick your favorite. You can use CS&T beads, you can use rainbow beads, other QC beads. Low to medium concentration, so you want to get 100 or so EPS, maybe a couple hundred. If you're on a platform that has a window extension, you're going to slam that down to 0 because you want to lose signal as soon as you aren't in the perfect place. Make a histogram to look at fluorescence off of each laser. You can do it for every channel you have or just one off of each laser. And when you're running the beads, adjust the display to be about 1/2 to 1 times the event rate. That way, when you make a change, it shows up. You see that histogram moving in real time instead of waiting for it to cycle through 10,000. And play with those laser delay numbers. If you bump it up a little, does the signal get better or worse? If you drop it a little, same thing. And find the place where you get the brightest, tightest peak. And that'll be your new laser delay. So this, you can play with these for troubleshooting if you're like, hey, is my delay off just a little bit? This is also a really nice way if you have to swap between nozzles fast on an ARIA platform and you don't want to sit through CS&T, you can redelay it yourself. And make sure you write down your starting points, screenshot them, make a note, whatever works best for you. That way, if you get too far off, you can come right back to something that's close. What if everything is failing? Right? The whole thing, everything's terrible. No baseball bats. I do have a rubber mallet and its name is Plan B. Okay. So think about if everything is failing, we're going to start thinking about very common factors. So fluidics and beads are pretty common to all of our lasers. So if we've got something really gnarly going on in the flow cell, but give it a good clean. If we're deflecting the whole core stream, then it's going to hit multiple lasers and not just one. Just one is usually it's slowed down from something. So again, soak and clean. Beads are also something that are going to be the same going across each laser. And they are incredibly robust until they're not. So if you're using beads that you made a few days ago or a week ago, maybe try making new beads just in case something happened to that tube. Make sure you're running the lot. You've got the lot number on the bottle. Is the lot number in the tube? Is the lot number you told the cytometer? Because if it's referencing a file and you've got a different lot, you're going to have all kinds of problems. And then, like, if there's any chance your beads could have become contaminated because there was some bleach on the outside of the sip, then now that ended up in your tube. Yeah, and it's easy to make new beads. So do a clean, make new beads, and then see if you're still failing or if it's made any progress. Who's going to yell at me about time? It's time. If you can't see any events, I won't make you guys go through the whole list. Yeah, that one's fun. We get that periodically. So start at the beginning. Anywhere in that pathway could be the problem. Again, lasers on, trigger laser on. Is it actually on, even if it says it's on in the software? You can always override the interlocks and take a peek. If you think your blue might be dead, if you swap your trigger to another laser, can you get signals for everybody else back? Do you see blue laser light? Thing. Did you load beads? Is the instrument taking up samples? So what if you have a fully blocked sample line? That could also cause this, and has. So load some water on high. Do you see the volume going down after 10, 15 minutes? No? Well, we've all been there. You can try cleaning it, but at that point you may just need to change your sample line. And you can either change them piecemeal if it's a multi-part sample line, or you can do what I did the other day on a sorter, pull it out of the pinch valve and just start taking pieces off until it sprayed sheath fluid everywhere. And I'm like, got it. Then replaced the rest of it. Okay. It's effective and quick. So cleaning, changing the sample line. Again, checking your lasers. Occasionally you can see beads in an experiment, but not in QC. At that point, just reboot because it's having an electronic hissy fit. I've got one that will periodically show nothing until it's had a couple of reboots, and then it's perfectly fine. Other potential issues that I do want to just quick highlight, especially because Matt kind of touched on them. High background in a channel. You're not necessarily going to catch this from instrument manufacturer QC. This is one of the reasons that I do recommend having a secondary multi-peak bead QC. We had, back at Wisconsin, we had an LSR Fortessa installed, and we were running QC, and we just were having a hard time seeing some APC signals. Things didn't look quite like we thought. We ran these 8-peak beads, and like the negative was really high. So for whatever reason, the filter set we had in there was getting laser light into it, which was bringing up the background and causing problems. So we put in a new filter set, and now we had reasonable APC again. So looking not only at where the positives are and how they're behaving, but where those negative beads fall can be informative too. Yeah, again, you see something really strange, sometimes a reboot will fix things. Stuck? What kind of resources do you have? Sometimes maybe you're newer to the field, or it's just you in the lab and it's 9 or 10 or 11 at night. You may or may not have other flow core staff around you. FSEs, instrument manuals are hit or miss, internet searches. But I do here want to plug the Flow Cytometry Discord. There is an entire channel devoted to getting instrument help. So if you join the Discord, and you can associate yourself with a role, or you can ping specific roles. So if you were having problems with an Aurora, you could go into instrument help, type @Aurora, and it would notify anyone who had signed up to be notified about Aurora problems. So this Discord community started by Dan. I asked him if he wanted his headshot or his Glyphka party shot. He chose Glyphka. So that was Dan as president of Glyphka. It's got a lot of people, and it's a global audience. predominantly US now, but not entirely. So you're going to have people in multiple time zones. You have people with buckets and buckets of knowledge. I have seen people taking apart sorters and pointing at circuit boards and where things need to go. So if you're stuck and you want additional help, this is another resource for you. We are generally friendly. None of us bite. It's online anyway, right? I don't think I'm a golden retriever quite, but not too bad. So yeah, this is another great resource for instrument help. You may notice there are also channels for assay help and data help. So if you're stuck on how to run something, reagent questions and analysis questions, this community has been really supportive and it has taken off quite quickly. So the QR code should get you signed up if you're interested. You can also, I don't know if you can search Flow Cytometry Discord. I never tried. You can also send me an email and I'll help you get there. So in summary, characterizing your instruments is important. Tracking these critical values can help you see problems before they're problems and give you clues to what's wrong when something inevitably does become a problem. If you're faced with a problem, take a deep breath, think through the system and where things could go wrong to cause what you're seeing. Soaking and cleaning is darn near magical. I highly recommend it. So if you're not sure, that's a great place to start. It also builds in a break of time to think. And the more troubleshooting data you can give to your engineers, the better they can also help and maybe get you fixed it faster. Because if you can fix it yourself, that is better than being down waiting for somebody to get to you. So here are some more resources if you're interested in screenshotting a few more. Thanks. Oh, yeah?
Yeah, soak clean and then power cycle. Well, it's soaking, and then bring it back.
Oh, there we go. Yeah.
Yep. So,
Yeah. The two birds with one stone.
exactly.
Clean it and reboot it.
I know, it's hard. Okay, just a quick question, actually from Matt, actually right here. That's okay. Question. When after resuspending hard dyed beads, do you have any like? I feel like beads can deteriorate in our hands with certain exposure to buffer. Is that universal? Any thoughts?
So, okay, so the exact question that you have is, after your beads are resuspended and diluted in something other than what's in the bottle,
Right.
do you experience deterioration? It's entirely possible. What is inside of those bottles varies typically, but it is normally an isotonic solution of some kind with some sort of a preservative. The most popular being sodium azide or was it phenoxyethanol, I think is another one that's kind of popular too.
Yep.
It depends really on what the beads are being used for. Going back to again, one of my recent efforts was figuring out a QC bead that could be used for a nanoscale instrument. And it's interesting when you get real low, you start to see some interesting artifacts, even of like preservatives.
Yeah.
So I would say it's entirely possible. I tend to try to make as little as possible if it isn't if it's not in a ready to use format. I would make as little as I need.
Yeah.
You can sometimes slow down, the degradation by refrigerating those mixes. But there's also a non-zero chance that they're going to aggregate with a different sort of kinetic, and in resuspending them after that aggregation can certainly degrade them. So I try to keep them as close to the original bottle as possible.
We've also seen weirdness if you accidentally grab the DI water bottle instead of the sheath bottle. So resuspending with sheath rather than DI water is important,
Yeah.
and making sure there's no accidental bleach.
So fun fact that we found out the hard way is that if you are using a bead that's like a functional bead with specifically with a carboxyl group, and you resuspend those in, I believe it's PBS, they can aggregate, and they can aggregate quick, and in places that it's very, very hard to get them out of. We're talking destructive reconditioning of a flow cell kind of places. So my suggestion is that not everything that you resuspend in is is universally applicable. So my general rule has become water for beads and isotonics for cells, because you can almost always, the beads will almost always survive water for some short period of time. So, okay, I'll get out of the way.
All right. Well, thank you, Rachel and Matt. Next up, we have some lovely gentlemen here to tell us about clustering your data. So their talk is titled Cluster Me This, a practical guide for working with automated cytometry tools. And that's going to be done by Jack, Nico, and Jeff. Now, Jack, he likes science, but the LA Rams more. So it's important to know. Not so sure they've ever really accomplished anything, but in his scientific career, more importantly, he focused on complex biological problems. I lost my thought. Yeah. And he worked on some really hard stuff. Sorry. The IKK complex and NF kappa B. According to him, he later secured parole out of those labs, and got to work on more fun stuff like neoplastic anation. Yeah. And then he, sorry, got a lot of experience in cytometry and microscopy and molecular biology. More recently, he spent 10 years working in the titans of the field. Many of you might know him from previously working for BD and FlowJo, but now he works for Metaphora. Close. That was close. Nico, also an expert in flow cytometry. Big surprise there. Did his master's at the University of Paris. And then he worked as a flow cytometry SRL manager at the Baylor Institute for Immunology, and then joined UT Southwestern in Dallas to be the director of the facility there, where he worked with over 120 laboratories. In 2021, Nico moved to industry and decided to join BD FlowJo as a senior application specialist and multi-omics specialist, and became the informatics solution leader in 2024. Hey, fancy. He is also recognized as a member of ISAC for a long time and a longtime member of the FlowTex committee. And then we have Jeff here. He also worked for many years in a core at Northwestern, and then spent 12 years working with the high parameter analysis tools as an application scientist at various softwares and instrument vendors. But now he's the senior application scientist at OMIC, which is part of Dotmatics. He's a persistent advocate for data quality and is passionate about educating people in finding approachable strategies for high dimensional analysis, which, we're all doing high dimensional stuff now. So we should listen to what they have to say because it's going to be useful.
So not for next year when there's 3 people, make shorter bio. All right, so when we decided with the committee to talk about data analysis, the first idea was for me to talk about data analysis, but I didn't want to be commercialized because really FlowTex, the idea in FlowTex is really to educate people. So I invited my frenemies, as I call them, with me for this presentation. So it's really going to be software agnostic. We're just going to give you the general idea on how to analyze the data for hyperparameter analysis. We're not going to talk about our solution, but just the big concept. So before we start about going for the workflow and the concept, we need to know what is a data analysis. So data analysis is a concept to unify statistic, data analysis, informatics, and other related methods in order to understand the analysis actual phenomena with data. So we use theory and technique from many different fields, computer, statistics, information science, and the domain that we study for us, flow cytometry, to be able to do the analysis. So why do we need data science? So like you saw this morning with the panel design presentation, the number of fluorochrome exploded for the past few years. We also make big improvement in the flow cytometry field for monoclonal antibody, and instruments now have 50, 72, whatever detector on it. So this results in a big increase in a publication with many fluorochrome in it. So we have from OMIP 60, which was the first one at 30 color, to the last one from the Frederick, and we have Peter Medge here about it for the OMIP 102 with a 50 color panel. So when you get that many columns, yes, you can still analyze your data using your biaxial plot and gauge for your population. That's feasible, but you need to know exactly what you're looking for to be able to find your population. So when I started flow cytometry, I was doing at that time what's called hyperparameter flow cytometry, which now seems ridiculous because it was an 8-color experiment. So to analyze this 8-color experiment, because I was a grad student and didn't know what I was looking for, so I had to look at the 28 plots to make sure I find my population of interest. It was taking me time, but nothing too crazy. Now, when we talk about hyperparameter analysis, we talk about 20, 30, all the way to 50 color. So here's a quick example of a 32-color experiment. If I want to look at all the 2x2 plot combination, I need to look at 496 dot plots. So if I spend 2 minutes per dot plot to define my population, I will spend 17 hours to gauge on this experiment. And I'm sure you'd rather do something else than gating for 17 hours. So we have to find another way. And that's where data science and use of algorithm came in play to analyze this data. So we went from biaxial plot to ends parameter hyperspace with t-SNE, UMAP, etc. That we're going to talk. So we can track really the uptake of the use of data science in the cytometry field by just looking at the number of citation in publication. And we can see over the year, using those algorithms really cranked up, and now is getting more and more. This stopped in 2019, but it's getting even bigger in the early days. And we can see that those publications are in every journal. They're not limited to only cytometry part A, but you can find it in Nature and every major journal. But data science is not only a tool that we use, it's also a field. And you can see that every year you have new algorithm that's been released. So you can see that average is about 6 to 8 algorithm, and they use different type of language for coding. The main one is R. So if you want to develop something, probably use R or know how to code in R, but MATLAB, Python, or C++ are also used. Most of those algorithms never going to be used. They're just going to be on one publication and never be used by the field. But you can see sometimes one of those algorithms start picking up and be used widely for the flow cytometry field. So TSNE, UMAP, part of those, Phenograph, EggShift, and others. But all those algorithms can be really classified in a different category. So I put 5 categories here. The first category is data QC. Second category is dimensional reduction. The third one is clustering, for visualization, and fifth, identification. And after I put another one, which is really specific to that algorithm. So first, we want to QC our data to make sure that whatever we put into the algorithm for dimensional reduction and clustering is clean and ready to go. If you put garbage in, you're gonna get garbage out. So that's really overlooked usually topic, but it is a really important topic. So what does an algorithm workflow looks like? So here's a general idea of a high dimensional workflow. So data QC first, annotation, concatenation, second, dimensional reduction, clustering, phenotyping, and comparison. So within the data QC, the first one I'm gonna mention, I'm not gonna go too much in detail, is the normalization. So normalization is the idea to remove any batch effect. So when you do a longitudinal study, you're gonna run different samples on different days, sometimes on different instruments, sometimes even on different locations, different sites if you have a collaborator. And with Paul, John, Maria doing the staining and everything, you may have a batch effect. And this batch effect, you're not gonna be able to compare the data between different days. So you have algorithm to minimize this batch effect. We have Cytonorm, MNN, Cycombine, and FlowStat. They work in different way. Really here the idea is to make sure that the data is comparable between all those different type of samples. Ideally, don't use them. So ideally, try to make sure that everything is normalized between your instruments. It's feasible. It takes time. Really is really the last resource to use this type of algorithm. I cannot go in data QC without talking about composition. So your data need to be well composited. You have many ways to calculate your composition. You have the traditional method. You have the auto-spill method. You have the spectral or the mixing method. We had talked about autofluorescence in this FlowTex. So of course, I count for autofluorescence to make sure that what you see is real. Transformation. So transformation is a step that's usually way overlooked. So it's really important step. What the algorithm's gonna see is what you see with your eye. So make sure that your scaling is appropriate. So make sure that you really see the negative population and positive population when you look at your data. Also, choose the type of scaling that you want. You have many type of scale. You have the logical, the bioexponential, arsenate. So really pick the one that works the best for you. Now let's go to the real cleanup data. So the first cleanup the data is just to remove any unwanted event. So you have 2 steps in this one. You have the automatic way to remove any air bubble, any clog, basically any issue that happen on the instrument. And after you have a second step that will be manual, is remove any event that you don't care about. So for the automatic, there's a few algorithms available. You have FlowClean, which was the first algorithm that existed to remove those problematic events. You have FlowAI, Peacock, FlowCut, and the MetaClean. They all have pros and cons and work slightly a different way, but at the end, they all do a good job. So FlowClean, the way it works, is gonna bin the data in small time bin. It's gonna split the data in half, positive and negative for every parameter. And it's gonna make sure between each time bin, the percentage of the positive and negative doesn't change drastically. If you see any drastic change in between each time bin, it's gonna say something happened on the instrument and it's gonna remove that time being. FlowAI is going to look at your flow rate check, make sure that your flow rate was consistent over time on your sample. It's going to look at your signal acquisition, so basically your MFI, making sure that it was consistent over time, and it's going to look at the dynamic range, or where your instrument worked the best, so within the linear range of your instrument. Peacock, same thing, is going to create small time bin. It's going to do a flow sum on it. It's going to make sure that you get the same cluster on each different time bin. The big difference between FlowAI and the other one, so Peacock and FlowCut, is that FlowAI is on raw data, Peacock and FlowCut is on composited data. FlowCut is mostly an evolution of FlowClean. Same thing is going to split in small time bin, but it's going to look at 8 different mathematical calculations to make sure that you don't have a significant change between each time bin. And last one, MetaClean. If you want to know a lot about MetaClean, talk to Jack. But it's basically an evolution of FlowAI to clean up your data. All right, so let's do a quick comparison between each of those algorithms. So we have FlowAI here on top. No, sorry, you're not going to see that online, so I'm going to use the laser pointer right there. So here we have FlowAI without the flow rate. We have a FlowClean, FlowCut, and Peacock. And really, in this slide, what interests us is the red line, so the accuracy of the algorithm. And you can see that FlowClean was kind of bad in this assay. So they use, need to go back, they use a FlowCap for data to check if the data, if the bad event were removed efficiently by the algorithm. So if there were, and the accuracy is really high. If they're not, the accuracy is low. So we see that the flow rate, the FlowClean accuracy was at 53%, so not really accurate. Every other algorithm, you're above 90% accurate. So except FlowClean, which you can see this age, which was the first one, yeah, use any other algorithm and just make sure that you know how to use them, how to tune this algorithm to get the best result. All right, so now that the data is clean, that we remove any issue that were coming from the instrument, now we want to remove any unwanted events. So of course we want to remove debris, so we do a cleanup gate. We want to remove doublets, so we use the diagonal versus A versus H, and we want to remove any dead cells and maybe lineage positive if you have a lineage gate. So that's the minimum. You can also start really zoom into your population instead of just doing the leukocyte, like in this example. We can see all the different cell population, monocyte, granulocyte, CD4, CD8, and NK. If I'm only interested in CD8, here on my CD8 population, I probably see only 5 or 6 islands, or potentially only 5 or 6 different cell population. If I start removing the cells I don't care about, so granulocyte, monocyte, B cells, NK, and CD4, and only focusing on my CD8 population, now I can distinguish way more different CD8 populations. So I have like 15 to 20 different islands, so potentially 15 or 20 different CD8 population. So really where you stop is depending on your scientific question. You may have to do both. Maybe you have no idea what your treatment or what the patient is going to be. So you're going to do your analysis on everything. Maybe you realize that it's your CD8 that react to your compound or whatever, and you're going to redo the analysis, only focusing on the CD8 population. All right, so now let's go to the next step, which is annotation, concatenation, and downsampling. So annotation is a big battle for me on annotation. I think a lot of scientists don't put enough annotation in their experiment, and it's really annoying for us. I think I will talk for the 3 of us, because a sample 1, tube 1, when you send us that by email and ask us, did my experiment work? Can you help me with the analysis? Sorry, but we have no idea what you did, so we cannot help you. So you have to send us your lab notebook and everything for us to be able to. So annotate your data. I know it feels like a waste of time for you because you think you're gonna remember what's a sample 121 from that day. I guarantee you that a month or 6 months from now, when you're gonna have to publish this data, you have no idea what you did on that day. And best case scenario, you kept your lab notebook really well and you can figure it out. I was not this type of person, so sometimes I had to redo the experiment and wasting time and more money. So annotate your data. Personal advice. All right, so down sample. Down sample can be for two different reasons. First, because you have so much data, so if you put it on the algorithm, it's gonna take forever to run. 24 hours, 48 hours, it happens. But most likely, it's just to make sure that one condition doesn't overpower another condition. So make sure that between each condition, you have about the same number of cells. And last step is really the primary difference between your standard analysis and this high parameter workflow is the concatenation step. So this concatenation step is because of the nature of this algorithm. The first step on any of those algorithms is the seeding. Seeding is random, so every time you run the algorithm on one sample, that seeding will be different. Doesn't mean that the result will be different. It just means that, for example, on the t-SNE, your CD4 population will be at a different location. Or if on the clustering algorithm, your cluster ones will not be the same, will be on one day a CD4 and another day another cell population. So to mitigate that issue, we concatenate all the sample into one big file. We run the algorithm on this big file that contains all the sample. And after, we just split back our sample and analyze so that. So you have virtual or actual concatenation, depend of the software, the tools, the pros and cons on both. And we're happy to help you. And now I pass the torch.
All right, thanks. Okay. So that was the first piece of the analysis kind of puzzle here, and that's how do you prep your data to be used effectively for the rest of it? And I can't overemphasize this enough. It really is important, like Nico was saying, in many different ways, to spend the time on the front end so that you can be effective using the tools that I'm going to talk about now for this section. So if you have any questions about what that might look like for you, all of us are happy to talk about steps you can take to clean up your data, to annotate your data. And we're also, I mean, honestly, we're also happy to talk about the steps I'm going to cover now. But I just wanted to put that out there. It's really important, starting with nice, clean data. All right, so we're going to be talking about dimensionality reduction and clustering and a few other kind of miscellaneous topics right at the end. This is something that I really enjoy covering. I'm going to make a lot of analogies here, so kind of buckle up. You're along for the ride. I'm going to start with dimensionality reduction. This is something that has been in the field for a long time. If you look at papers, and PCA has been around for decades. At this point, it's bread and butter of a lot of genomics work that goes out there. And now in cytometry, we're seeing it all over the place. I've been asked before, I had a picture of a t-SNE up on a poster I was presenting at a conference, and somebody asked me now, it was probably 7 or 8 years ago, they asked, hey, that's a really cool tissue section. What is that? And at the time, I was like, oh, what? But it actually did kind of look like a tissue section. Now, these days, though, people find it more familiar. So the field is kind of getting into this more computationally assisted space. But I still am on this talk going to kind of target this to make sure that everybody has a reasonable grasp of what these tools are and how you can use them. So analogy number one, dimensionality reduction. These are great tools, and there's a lot of flavors. We'll cover them. But I like to talk about it this way. It's these algorithms' goal to take many dimensional differences and kind of distill them down and give you the best and often two-dimensional representation of the many dimensional differences that are in your tubes across all of your events. I often will talk about things in relation to PBMCs. So if you don't do PBMC work, apologies to the neurobiologists, et cetera. Generally, it's a convenient framework to talk about these tools. And so typically for dimensionality reduction tools in cytometry, people will see different islands. Each island will be a different cell subset, and there'll be some heterogeneity inside of each island. There'll be little lagoons or archipelagos or whatever. I like map terminology. So we'll talk about those once we get there. But that's, again, the best kind of two-dimensional representation of a many-dimensional object, your data. We can do this in other ways too, though. So a many-dimensional object, well, I mean, three's not many, but like an apple is a three-dimensional object. And I've made here a two-dimensional representation of a three-dimensional object. And it's fine. You know what this looks like. Everybody would be like, yep, that's an apple. But is this a better two-dimensional representation of a three-dimensional object? And I'd like to think so because I made this slide. But I mean, generally people would say, yeah, it kind of depends what your goal is. But yeah, that's a better representation. This is a photograph. Is this the best two-dimensional representation of an apple? I don't know. I mean, it's a pretty good one. Another way I like to think about this, though, is going from a globe to a map. You're taking a three-dimensional object and reducing it down to 2 dimensions. And there's lots of ways to do this. This is, ripped mostly from XKCD. It's a comic near and dear to my heart. But basically, there's lots of ways that you can go through and project a three-dimensional globe onto a two-dimensional map. Some of them are really familiar. Some of them have Russia and Alaska touching, which is, unique. It's not something people see that often in maps. Some of them, have geopolitical implications about what they emphasize or how big the land masses are, et cetera. And the same is true with all of the algorithms we're going to use. You can see I've kind of salted those in there. Each of these algorithms is a way to project your many-dimensional data into just a handful of dimensions, whether it's actually something like UMAP or t-SNE or kind of something more like clustering. We'll get into what the difference is a little bit later. But broadly, these are tools, and they're just choices you can make. There's not a single correct one. There's not a single incorrect one. It totally depends on your problem. So if you have questions about what might be best for your data, the best is to talk to an expert. So whether it's one of us, whether it's people at your own institution, whether it's the Discord, and I have a shout out to the Discord at the end of this talk also. Those are great places to start. So given these projections of a map, is this the best picture of an apple? No, probably not. But it's another way to look at an apple. And so, we got cut off there. And so basically the goal of dimensionality reduction algorithms are to give you kind of a good low dimensional representation of a high dimensional object, your data in this case. And each of these islands are a group of similar cells. The more similar a cell is to another cell, the closer they'll be. Although just big disclaimer here, in these kind of algorithms, these nonlinear dimensionality reduction algorithms, I'll touch on that in just a second, distance between the islands does not mean very much at all. I'd say vanishingly little. What is important is if two events are in the same island, they're very similar. If they're in different islands, they're different, but you don't get information on how different they are using things like t-SNE and UMAP. So there's lots of different tools out there in the field. t-SNE and UMAP are two of the most common in cytometry. Things like TriMap, and this is, in Peter's talk earlier, he said, mathematicians are lazy. They want to take a solution that's already been kind of put out there in the field and then repurpose it. And then we see this pattern in lots of places. So people will take an already established algorithm and then riff on it to better suit it to their particular problem. And so again, we'll see more in just a second. But essentially, t-SNE and UMAP are two of the most common ones in cytometry. There's lots of other ones out there, though. And I'd say try as many as you can and see if it works for your data. I'm pretty pragmatic when it comes to analyzing cytometry data. Does it work? Can you find your populations of interest? Can you communicate your populations of interest to other people? If you run a brand new algorithm that nobody knows about, you then have to take that burden of describing what that is to other people, whether it's your boss, the reviewers, an audience like this. It comes with some responsibility. So using common ones is a good idea. Really quickly, and these are going to be things that we look at kind of throughout. I'm not going to read the slide, and we'll see a few other slides like this in this presentation. They are useful definitions or useful settings, ways you can tune these algorithms. Because this is being recorded, I want to make this information available, but also don't want to just like kind of drag you through the mud listening to me talk about this stuff. But I will take just a second to cover these things. Distance metrics are really kind of useful to think about if you want to get more into data science. But as far as cytometry is concerned, a Euclidean distance metric is a great default. Kind of start there if you're ever presented with this option with some algorithms. Nico already covered random seed for a little bit. Basically, it's kind of what random choices get made. If in an algorithm that you're tinkering with, you have the option to set the seed. If you set the same seed with the same settings, the same data, you'll get the same result. This makes people who are worried about repeatability, it makes them kind of feel warm and fuzzy on the inside. You can get the same answer every single time. Practically, though, I haven't seen a lot of people need that. There's a handful of other ones. The one I did want to talk about here specifically is linear versus nonlinear. PCA is a linear dimensionality reduction algorithm, and without kind of opening up a whole new can of worms, basically what that means is the distances between the islands or between where your events fall on a PCA plot are very meaningful and can tell you quantitatively how different things are in many dimensions. It turns out PCA is not that great, though, for visualizing flow data. For nonlinear algorithms, things like UMAP and t-SNE, these are algorithms that are great at visualizing flow data, but the distance means a lot less. If you have questions about what specifically that means, again, happy to answer that later, but just a special note to call out. Another concept to talk about is local versus global structure. So I mentioned these are just tools. They're helpful tools, but they're, they can be used lots of different ways. I liken it to like tools in a workshop. You could probably, you could use a screwdriver like a chisel, and I can't tell you how many times I tried to do that as a kid. My dad would be like, "What are you doing? That's not what that tool is for." It kind of works. You can kind of do it. And the same is true with these things. A lot of these tools can work for a lot of different purposes, but some are better suited than others. And so, for instance, this local versus global structure, these are basically concepts of like what you want to focus on. If you want to focus on local structure, t-SNE might be better. And local structure is essentially the heterogeneity inside of a particular cell subset. So if you want to emphasize the difference between your RA positive and RA negative Tregs, those are small differences driven by just a handful of markers in most panels. Then you can use something like t-SNE, which is better at emphasizing local structure. It doesn't really separate your cell subsets that nicely. It's not as clean of a visualization, but it will give you more heterogeneity inside of a particular space. Global structure is what UMAP is a little bit better at. Big, nice, clear spaces in between your islands. However, it kind of smushes that CD4 island kind of together a little bit more. You get less heterogeneity displayed. And so UMAP is good at showing a nice clean picture, but is less good at emphasizing these kind of very small biological differences. So if you're looking for batch effects, maybe try a t-SNE. If you're looking to have a nice, clean, really easily interpretable graphic in your figure, for instance, maybe try a UMAP. Feel free to play around, though. There's lots of people that publish with t-SNEs, and there are lots of people that use UMAPs to look for batch effects. Both are all right. So here's another one I'm not going to read through, I promise. But the takeaway here is that there's lots of different versions of t-SNE. People have found the initial one to be potentially useful in cytometry, but slow and not very well suited. And so they've worked on it, tinkered with it, and there's lots of different versions now that are much more well suited for use in our particular space. Flow data is a really interesting and kind of convenient structured type of data to use a lot of these machine learning tools, But a lot of these tools were not made for us. It may seem that way. I like to think so sometimes. But things like TSNE, things like UMAP, things like self-organizing maps, k-nearest neighbors clustering, those are data science tools that people use all the time in many other fields. And we're just happily adopting them into our space. There's lots of versions of TSNE. Some of the newer, kind of easier ones to use are things like OptSNE, which kind of knows when to end itself, which is nice. So you don't have to tune it a ton to get it to work. FitSNE, which is a very, very quick way for TSNE to look at your data. And again, if you want any details on any of these hyperparameters or settings, come by and ask us specifically what you want them to be. UMAP here is, again, kind of a better focused on global structure, and will allow you to, kind of get a broader sense of all the different cell subsets that are in your particular experiment. I mentioned this is a brief aside. I mentioned hyperparameters. That's a different kind of technical term for the settings you use to tune an algorithm to better suit your data. A lot of the default settings in a lot of our software are kind of well suited for cytometry data. However, it can be better. This is something just keep in mind. Just because an algorithm spits out an answer the first time you run it doesn't necessarily mean that that's the best answer or the only answer. You can change these settings, whatever, I mean, basically all the ones that I listed here, the iterations, perplexity, learning rate, nearest neighbors, minimum distance, all these things can be changed to better suit data. And it just takes essentially sitting down, running it once, saying, hey, did that work? Did it not work? If not, what can I do to make it better? And tinkering with those settings a little bit. Yeah, basically the takeaway here is there's not a single correct setting. Basically, if it works for you, if you're able to tell your story, if you can kind of pick it up and run with it, communicate your results well, that's a good result. All right, so now into clustering. This is different from dimensionality reduction. And actually, you know what I'm going to do? I'm going to hop through this because, oops, I want to talk about this really quickly. What's the difference between dimensionality reduction and clustering? This looks like some Halloween candy. This is my favorite analogy. So I don't know if you've ever been around kids that go out trick-or-treating, or if you've done it yourself as a kid. When you come back, you dump out your candy on the kitchen table and you're like, yeah, candy. And then immediately it's a compulsion. This is my daughters from a couple years ago. They start sorting out candy into little islands. That's like dimensionality reduction. Just taking that pile of candy you dumped out on the kitchen table and putting the similar things next to each other. That's a very effective analogy for what dimensionality reduction is. Clustering is technically different. Clustering is if your parent gives you some Ziploc baggies and says, all right, now take your candy and put them into these 10 baggies. Maybe you have 12 piles. You have to make the choice. Does, do my M&Ms go in with my Reese's Pieces? Because they're both small and round and candy coated, but one is peanut butter and one's not. And do peanut M&Ms go in that bag too or not? You got to make hard choices. Putting your populations into discrete groups, that's clustering. Just having islands kind of arranged on a kitchen table, that's dimensionality reduction. You're not making hard choices with that. With that context, we'll dive back into clustering. Should have put that slide first. So basically, clustering is a way to put similar cells into populations with each other without knowing or without having to know the phenotype of those cells. So I know how to define naive CD4 positive T cells, but what's positive for 16 or kind of dim positive for 16 and positive for HLA-DR and positive for RA, and maybe you don't, it's 14 negative, and you're like, I don't know what that cell is. It could be that could be anything. For these interesting, weird populations, you don't have to know how to define them, but it might still be good to keep an eye on what's going on with those. And so clustering is a great way to, even without immunological knowledge, you can still get cells that look similar to be in the same group and then track that across your experimental conditions. So there's lots of ways to do this as well. fewer typically for cytometry than dimensionality reductions. But here's some common ones. Basically, an unsupervised algorithm, and this is a kind of an important technical note. Unsupervised in this case means something very specific. It's not just humans don't have a lot of effect on what this is. It means that the algorithm itself doesn't know what experimental groups your samples belong to, and doesn't have to know in order to work. So unsupervised has a very kind of narrow technical meaning. It doesn't mean that humans don't have an effect on the outcome. I set up an algorithm a certain way. You know, Nico or Jack, they might set up their algorithms with a different set of hyperparameters or starting with a different starting population. And that will absolutely have an effect on the outcome of that particular algorithm. I set up things because I learned how to do, you know, spade from Jonathan Irish. Other people learned how to do spade from other people. And that is absolutely going to have an effect on the outcome. So it's a quick tangent, but just something to keep in mind. Unsupervised has a meaning, and in this case, clustering, dimensionality reduction, these are all unsupervised machine learning algorithms. Okay, in this case, though, we're going to dive into the options here. So this is one that is relatively new. It's a newer one out there. Park is essentially an algorithm that you don't have to tell it how many clusters you expect. It will try to find that structure in the data. It's very fast and scalable. It uses, if you're interested, the Leiden algorithm for community detection. This is kind of a new, cool way to look at clustering. But if it doesn't find your particular population of interest, the first go through, that's an issue. You have to go back in and tinker with some of those settings in order to be able to find those Tregs or whatever, because that's known biology. For all of these algorithms, regardless of the type, it's important to track it back to how do I understand the biological issue, and can I rectify that with what I'm seeing here in front of me? Just because an algorithm did it doesn't mean it's right. It's just a tool, and it's a good idea to double check that. XShift is another kind of slightly different way to tackle clustering. It uses k-nearest neighbors. It's fast, does not require you to input a specific number of metaclusters. It's good at finding small and rare populations. But sometimes, again, if it doesn't find your small and rare population, it's going to take some tinkering to get it to pull that out. And there's no kind of default visualizations. It's up to you to kind of overlay those clusters on a t-SNE or a UMAP or 2-parameter dot plots, things like that. FlowSum is a really common one in the field. It's common for a reason. It's really quick. It's great at clustering. One of the switches, though, here is that you do have to tell it how many metaclusters you want it to split your data into, how many pieces of pie you want it to cut for you. And that requires typically a running it once. Hey, can I find my smallest population of interest? If not, all right, let's walk it back. Let's try it again with more or fewer. And so it's a slightly different dynamic there. And then Metaflow, again, this is kind of the Metaphora proprietary algorithm. You don't need to give it a cluster or metacluster number. You can adjust kind of the depth after it's completed. So you can say, all right, what if I want to split my populations up more? You can go a little bit deeper and split it up that way. It gives you some kind of visuals or some projections. But it is proprietary to Metaphora. So if you want to try it out, go talk to Jack afterwards. So again, those are the kind of the big differences. So once you're done, and this has been kind of the space where the field is at for a long time, is all right, so I've got dimensionality reduction done. I'm clustering. What do I do now? Kind of the first step is say, hey, are these clusters good clusters? Did my algorithm do a good job of putting events into the clusters that are similar to each other? And so here, there's a couple ways to evaluate this, but it basically you're looking for homogeneity in the clusters. You want to make sure that you don't have both 45 RA positive and negative events in your lymphocyte subsets. You'd prefer that to be kind of only one cell type per cluster. After you have your clusters, they check out. You're like, yep, I did a good job. Way to go, me. The next step is to say, are there any differences between my experimental groups? Using your annotation that you have painstakingly entered in, you can now start to ask questions like, all right, what's different between my treated or untreated, or my responders and my non-responders. And there's lots of ways to do this. There's some computational methods like EdgeR. There are kind of box or violin plots. There's heat maps. There's a ton of different ways to kind of show, okay, now that I have these things clustered, what is changing between my experimental groups? And it's important, well, I think it's important, take what I say with a grain of salt, but I think it's important to look for differences first, because if you try and phenotype first and then find there are no differences, that's going to be really deflating. I know, because that's happened to me. So phenotyping is saying, all right, I've got these clusters. I know they're different between my two experimental groups. What do I do next? The answer is find out what type of cells they are. Why might they be changing? Kind of go into literature and figure that out. There's lots of different ways to phenotype. I'm kind of partial to this figure, and it's kind of overlaying events in a cluster on top of your total events. And each one of these columns is a different marker from your panel. So you get kind of a signature of what's going on. But there's, again, heat maps, overlaying on top of dimensionality reduction plots, like coloring them by expression level. There's tons of ways to do this. And we all have opinions. So you got your differences. You have your cell types kind of listed. Now what? Make it compelling with cool colors. If you go to this website, learnui.design, or this coolers.co, these are two websites to help you pick out really cool palettes. I had this, let's see, wherever it is, floating around for a while. This is the figure from OMIP 69. And this is, I used that learnui.design color picker to color this figure. And so you can have kind of thematic spectra around each of your different cell subsets. So I would definitely recommend taking time to make your figures look nice and cohesive and good. That's great. And then another thing you can do is to learn more. So these are some links to some blogs or some YouTube channels or online resources to pick up more about data science in general. Always a great idea to keep learning. And then, like Rachael already mentioned, join the Flow Cytometry Discord and ask us questions there because we answer questions there. So now I'll hand it over to Jack.
Okay, so my portion is just to kind of talk about the practical application or the use case scenarios where you would use these tools and the kind of the things to think about. So I think kind of along the theme of a lot of what's gone on today is you always have to ask the question, what am I doing? What am I looking at? And when it comes to data analysis, you want to make sure before you jump into using these tools to ask yourself the right questions about, first of all, how many samples am I running and what is the dimensionality of my data? If it's not that complicated and you don't have that many samples, I wouldn't even bother with these tools, right? Just stick with manual gating, get your answer, and move on. But if things start to increase in the number of samples that you're going to process, or that in the future that this experiment is going to be something that you're going to repeat over time, then the use of these tools becomes a primary component of your analysis pipeline. So I just put a bunch of questions here, that I think are practical questions to ask yourself before you get started. And the way I'm sort of tackling this portion of the talk is just basically from three basic questions, which is, starting with the number of samples, the number of dimensions, and whether or not the phenotype of the cells that you're looking at is known or unknown. But there are a lot of other questions that will influence which tools you are going to use as an outcome. So for example, what is the research setting? Obviously, if you're RUO versus clinical, it's going to involve different sets of tools simply because of the audit trails that you have to have in place in order to validate the study that you're working on. How much money are you willing to spend, right? Because some of these things, like Metaflow, for example, are in the cloud, and you have to pay a fee in order to use them, right? Others are free, like in R. And then other things. How do the outputs of my data from either TISNI or from the clustering algorithms, how does that feed into a downstream pipeline, especially if I have a large database and I want to somehow interrogate that legacy data and I want to say, see what happens to the frequency of my CD8s over time, or as a result of some perturbation that I've been working on for the past 6 months. So I'm taking it again from the top 3 cube kind of angle, and we're going to extrapolate those use cases, but just keep in the back of your mind, there are many other questions that could be asked that will influence the rubric that I'm going to be using here. So I think the way I'll approach this is kind of from the kind of a reverse Z, if you will. So as I mentioned in the beginning, like if you have low number of dimensions and there's a few samples, this is an easy experiment. So we're going to sort of ignore the bottom left corner over here. And then I figured I would approach this from the easiest sort of application to the hardest, moving in that direction. So the next sort of easiest application to go for is something that's relatively repetitive, has low dimensions, but you've got a high number of samples. And then we move into the direction of high dimensionality, but few samples. So I call this like the discovery experiment or your one-off experiment. And then when things start to get, really crazy, I extrapolate a bit further here, asking the question about, whether or not the phenotype is known or unknown when we get into that upper right quadrant, because then you can go in a lot of different directions. And there's I think a lot of basic things you have to ask yourself in terms of deciding what the practical pipeline is going to look like. So if we just consider that first, bottom right corner, this is like a screening assay. I would say here, don't even bother with clustering tools or dimension reduction, because most of these clustering tools need more than 6 dimensions in order to give you some valid output to begin with. Same thing with dimension reduction. And it's not going to be all that informative for low D data. But nonetheless, this is a true flow cytometric application where you would want to use some computational tools to help you with this. And so at the bottom here, I kind of recommend, Overton subtraction. So if you're looking to see, what is my percent positive if I'm doing some sort of a screen for antibody binding, for example, what are the percent positive that I'm getting from that? Well, you can use Overton subtraction or something called the super enhanced Dmax to evaluate the percent positives. And you can get some additional information like a chi-squared value to let you know how significant those differences are. Other low D types of screens are looking at cell cycle or proliferation studies. All of those tools, you can put into a template and you can get a result from without thinking twice. And if we go into the discovery, so this is that upper left, this is few samples, but high dimensionality. I would recommend here that you use dimension reduction, but kind of avoid clustering, because first of all, clustering, trying to figure out all those little baggies that the Reese's Pieces or the M&Ms go into can cost you a lot of time, especially if you don't really need a tool like that to be used to get the output that you're looking for. Dimension reduction in this case will work fine to just give you an idea of where the phenotypes are changing, and then you can chase that with manual gates. So I've given the example here. Geoff covered, I think, really well. I prefer t-SNE for looking at small phenotypic differences between the samples. If you're looking at trajectories, let's say over time, you want to see, let's say B cells that are not class switched. You want to see their development to a fully class switched B cell. Sometimes like a UMAP will help you with that because the trajectories of those cells will appear as little peninsulas from those islands. Okay, and then when we go to the upper right, this is what I like to call the everything, everywhere, all at once. But if we put this on a new set of axes, so we're still looking at low D versus high D, but now we're considering the next dimension, which is phenotype being known versus phenotype being unknown. There are some changes to the algorithmic setup that I think you would want to consider. So again, I'll probably approach this from what I would like to call easiest to most difficult, but in this case, we start in the upper left and move down here and then up here in this scenario. So for this one, for the templatable clustering, this again is the high number of dimensions, but your phenotype is known. And the assumption here is that you have a decent number of samples, so like 50 or 100 or more. In this case, a clustering algorithm like FlowSum would be good, where you have a fixed number of clusters. You tell it, all right, I know what phenotypes I'm looking for. Give me 20 clusters, and I'm going to observe what changes across those 20 clusters in my experiment. I also mentioned Metaflow here because you can adjust the numbers of clusters, so it's useful, I think, in that setting. Fixed embeddings. So t-SNE, for example, if you run it in a longitudinal study, it becomes cumbersome to have to concatenate files every month or every 6 months and then get a projection, because that concatenated file gets pretty large. I know that FlowJo has made some improvements there, so I think you can project into the same space now without having to reconcatenate the files. But EmbedSum was initially developed for that purpose, so that wherever your CD8s landed in a two-dimensional space, they would always end up in that same spot, regardless of where the file came. So I think in this scenario, fixed embeddings would be useful. You can also take the normalization setting there kind of as with a grain of salt. I feel like normalization in flow data is kind of a toss-up, because you're always running the risk of erasing a true phenotype that might be there. In this case, I'm kind of leaning towards go ahead and try normalization, simply because you know what phenotype you're chasing. So if you know what you're after, the normalization should actually help you, right? Because it'll remove those batch effects, and then the answer that you get in the end is going to be cleaner. Okay, but in other scenarios, it may not be so useful. So in this case, this is what I like to call the MRD monitoring. So this is relatively low numbers of dimensions, high sample number, so like a clinical, clinical panel, something like that. But you may have an unknown phenotype, or the phenotype changes. So the example that I've given on the right here in the two plots is coming from the FlowCap BAL data, where they're looking at malignant blasts in blood, and in the top plot here, you can see that they define these blasts on the CD38 versus CD10 axis. Whereas in the bottom plot here, you can see they're looking at CD45 versus CD10, because in that case, the CD45 was the more profound factor that was changing in order to define these populations. So in this case, you have a very interesting situation where that phenotype is unknown, or it's sort of bouncing around. And how do you make room for that, right? How do you get the clustering algorithm to actually monitor that and let you know? So one of the ways you can do this is, again, to use FlowSum with a fixed number of clusters. But in this case, you give FlowSum, you overfit the model so that you give it room to identify new populations of cells, but within a sort of a fixed framework. If you use something like Phenograph or XShift, you're going to get a bunch of populations that will find these different blast populations, but you never know which cluster that's going to be. So just from a practical standpoint, I feel that FlowSum, because the structure is predictable, you can just make a little extra room for it. And if that MRD phenotype pops up, it'll pick it up, and you'll see at least the frequency change that happens for that particular set of clusters. And then again, normalization is probably not a good idea here, because you have a good chance of normalizing out the biological difference. So you don't want to flatten out that MRD, especially if you're chasing 20 cells. Those can easily get merged into the next largest cluster. And I would say that as far as dimension reduction is considered here, again, small phenotypic differences that you're looking for might be more useful than a UMAP. So I would project onto t-SNE space in this sort of scenario. And then another tool that you might want to use is some form of ratiometric monitoring. So label all the phenotypes that you should have, right? And you see the balance, let's say, between CD19 positive B cells, and then your what do they call that? The hematogone population, which is also CD19 positive, but doesn't express some of the IG markers. So watching those ratios shift around can help you to identify samples that have problems in them. All right, and then the last use case here, which I have to say, like, I'm not exactly sure that the flow community is doing a whole lot of, where we have high sample number, high number of dimensions, and unknown phenotype. This is like a high-end kind of discovery experiment. But in this case, my recommendation here is either using phenograph or X-shift, again, where you're not fixing the number of clusters, you're just keeping the algorithm open, open to the idea that there may be something new there, or using something where the clusters are adjustable kind of makes sense. The clustering aspect of this to me is very difficult because, again, you're working with an unknown phenotype and you don't exactly know what you're walking into. So to direct the algorithm to do something in particular for you is a little bit, I don't want to say difficult to interpret, but it's difficult to interpret. Or it's going to take you some time to interpret what those results are. So what I find helpful in these scenarios is, again, same reasons to use t-SNE and UMAP. It's UMAP if it's trajectory, t-SNE if it's a phenotypic, a small phenotypic change. And then something else that could be really useful here is looking at correlation plots, where you see, how do the markers in a particular experiment here correlate with one another. And that'll help you to identify the phenotypes that you ought to be chasing, which will effectively feed back on the clusters or the clustering algorithm that you use here. And ultimately, it'll probably revert you back to something like this, where you will probably use FlowSum, but an overfit model of FlowSum to identify those new populations consistently. And then I think Geoff covered this, pretty well, but I always like to kind of give people the idea. These tools, I think at first glance can kind of seem scary, but the reality is just be consistent with whichever one you choose. So if you take anything from what we say here today, there's 1,000 different algorithms out there that you could use. Just be consistent with which one you use, because ultimately the differences between the two is how they slice up the data. So in the case that or the example that I'm giving here on the left, the first clustering algorithm, has a straight grid. The resultant populations that it gives you can see are the individual dots for each of the, let's say, major populations. And this could be maybe your, the red population here could be your monocytes. You get, 3 different clusters as an output, from that algorithm. But if you use algorithm number 2, suddenly you get some splits in the data that you didn't expect before. But regardless of which one you use, if you stick with that through your entire pipeline, it'll help you in the end, because if you chase all the different algorithms, it's going to cost you a lot of time. And then again, phenotypic differences here between t-SNE and UMAP. I think Geoff did a good job, so I'm not going to cover, I won't talk about that too much. And then just for your own interest, if you guys would like to know, I just did a little bit of research to figure out which clustering tools tend to be the most popular. So FlowSum is definitely top of the list. That's the big blue slice of the pie here. Phenograph follows that, at least for flow cytometry applications. And then I think it's Spade in the purple. But as you can see, the remaining clustering tools that are being used is relatively equal. And then for mass cytometry, that trend effectively follows the same sort of pattern, FlowSum and Phenograph being the winners there. And then as far as dimension reduction tools, a lot of people are publishing with UMAP over t-SNE. This was, the analysis here was done just looking at the last 4 years, so 2020 to 2024, to see what was published in a lot of journals like Nature, Cell, or Science, and their sub journals. The one interesting thing that I learned from looking at all these things is that people that are doing mass cytometry, and by default mass cytometry tends to have more dimensions on average than a typical flow cytometry experiment would, they tend to use clustering tools much more often with their data than flow cytometrists do. So a lot of flow cytometrists are using dimension reduction, but not so much in the way of clustering. And that kind of goes back to the example that I gave earlier. I don't feel like a lot of us are doing high D, multiple sample phenotype unknown type studies with flow. I may be wrong that there's a huge assumption there, but that's just what I don't know, what I found kind of interesting. Anyway, thank you all for allowing us to come out here and talk to you about high D stuff. And yeah, I guess take questions if we have time.
There's some questions from the internet. There's two from Daniel. The first is, when is Geoff releasing his podcast on data science for cytometrists? But the second, which maybe there's more detail, are there any big differences when doing analysis on data that contains morphological parameters from imaging cytometry? Or it would be interesting just to hear about using the algorithms on flow imaging stuff too.
I mean, I haven't looked at a lot of data that have, imaging parameters associated with them. So I would assume that like a lot of the stuff that was done with the S8, for example, that, is probably pretty cool, because I think that brings in at least five or six new dimensions in addition to the fluorochromes.
Way more than that.
Ah.
Yeah. Yeah. So with the imaging parameter, with the imaging parameter, you get more resolution. I have slides on that that I can present if you stop by the booth. But you get way more resolution on the population with the imaging added on the fluorescence. So, and that's what we're doing also after developing all those tools for.
Yeah, for the morphological parameters, totally cool to use those when you're clustering, but just make sure that they're scaled to match your other parameters that you're using. So a lot of the time, fluorescence parameters, if you're not transforming them, are like 200,000. If you put in like some sort of like moment or whatever, and it has units between like 0 and 10, it's not going to change anything at all about the clustering. You won't effectively be using that to separate your cell populations. And so either scale your fluorescent channels, which a lot of people already do anyways, so to arcsine H, and that's between like, -1 and 6 or something. And then that plays much more nicely with smaller scaled morphologic parameters. Or you can go in, do some FCS file surgery, and multiply, your morphological units by whatever you need to get you in the right neighborhood of your fluorescent parameters. But yeah, and then tell people you did that, and then you're in the clear.
There's another one online. Any suggestions on adjusting the signal range of each marker based on its expression level in a flow plot before dimension reduction and clustering? So I guess maybe the transformation? Yeah.
Yeah. I guess, yeah, assuming that signal range means something like transformation. Nico covered this earlier also, but make sure that you have the data well transformed, because if there's a split symmetrically around 0 because you have it poorly transformed, and it looks like there's two negative there's a population right below 0 and a population right above 0, the algorithm will think, oh, that's two clusters, even though both are your negatives. And so making sure the data is transformed correctly and then running clustering is a great idea. But maybe if I'm going to go like a little crazy here, I've talked about maybe adding in some sort of multiplier, because people choose the brightest floors for their most important proteins. And so people are changing the essentially the range or the weight that they're adding to particular markers. And it gets really sketchy though if you're letting people just like slide the importance of markers up and down, and therefore the units associated with those. So I don't think anybody's been brave enough to release a software that allows you to just change the weight of markers. But I'm not saying that there shouldn't ever be that. There should just be some like, guardrails or something around it. Great, thanks.
Cool, thank you guys.
What? Oh, yeah. There's a group photo tomorrow at lunch, so please come for that. And that wraps up the first day of FlowTex. Thanks to everybody for coming. We will start talks tomorrow at 9 a.m. Central. Okay, thank you.